I welcome all those who have just joined. We would be starting in more 10 minutes. We are waiting for the others to join as well. Yes, Nisar, you will have the session recordings. We'll share the recordings with you over mail as well. And also you can find the recordings on YouTube. We are streaming live. You can have just paste the link of the YouTube with in your chat box or in your notepad maybe, and you can refer it in future. Yes. And by the way, the Zoom link and the YouTube link would be the same for the whole day. So you need not worry. So Nisar, we have a criteria for the certification. My team members will let you know. You need to register yourself on the Omics Research Symposium registration page in order to get the certificate, because it's not possible for us to keep a note on who all is joining via Zoom. So please fill out the registration form. We'll share the details with you. Please drop your mail ID here, or my team member Ujasvi and Navya will tell you the criteria. Please register yourself so that we have your registration, and then we'll send the certificate. Thank you. I welcome all those who have just joined. We would be starting in 5 more minutes. Thank you for joining. Thank you everyone for waiting so patiently. We would be starting in few minutes, giving in time to others to join as well. Hello and welcome to the second day of Omics Research Symposium. This two-day event is designed to bring together students, researchers, faculty, and industry to discuss recent advances in omics data technologies and analysis. In this symposium, we will discuss the changing role of bioinformatics. We will learn about exciting projects in academia and industry, such as clinical, pharma, biotech, research, and even agriculture. As a result, you will learn about exciting projects in academia and industry where data analysis skills and solutions are urgently needed. The symposium discussion will be accompanied by a poster session and competition awarding prizes and giving a stage to students, faculty, and research teams to present their work and get expert feedback. You will also get a glimpse of international community working together to advance bioinformatics education around the world to make this field more accessible to anyone. It has now become increasingly clear that there is a huge demand in bioinformatics training, which has led to the establishment of Omics Logic bioinformatics training programs, with thousands of participants, tens of university programs, and local chapters in Asia, Africa, and the America, where dedicated teams interact with students and faculty. We will also hear from these teams about their achievements and vision as the program continues to grow. As the pandemic continues to keep us safely at home, we hope that we can make the most from this virtual experience. I'm sure there will be some technical glitches and the interaction will be very different from attending in person. And that is why I would like to start by addressing some common questions at the start of this program. You will find all the important information about the speakers, their talks, and the schedule on a symposium schedule page. The same website contains other information on poster competition, Zoom links, and panels where we will be able to ask experts about their thoughts on the topics we discussed today. Use the chat box at the bottom right corner of the screen to submit your questions for panelists or get in touch with our team for any other questions you might have. We will also invite you to review and share the content you will see here on social media. You can connect with us on LinkedIn, Slack, or Facebook. My team members would be sharing the link with you all in the chat box. You can also find us on the other social media platforms where we will be sharing updates, insights from the symposium, and also introduce our speakers. But let us focus now on the reason we are all here today. This second day of the symposium is dedicated to how can we get involved and make an impact in data and life sciences. So to introduce that, I would now like to invite Mr. Ilya Brodsky. Mr. Ilya Brodsky is one of the co-founder and CEO of Pine Biotech. He has facilitated and managed collaborations with researchers at Stanford UCSF, Louisiana State University Tulane, and other academic and pharma R&D teams as well. Mr. Ilya Brodsky oversees the research projects and manages the development of the prototype, preparation of the relevant data projects, and implementation of the system for the need of students and teachers that the project is designed for. Over to you, Mr. Ilya.
Thank you, Sonalika. And before we begin, maybe we could launch the poll. Sonalika, you will launch the poll?
Yes, just a second, Ilya. Can you just launch the poll after your talk?
Okay.
It's asking me to sign in. I'm not signing in from the official account, I think.
Okay. All right. Thank you, everyone, and great to be here today. And then thank you, Sonalika, for the introduction. So I'm excited to be here on our second day. And as Sonalika already mentioned, we will go on to speak about how one can get involved in bioinformatics in a more serious way to contribute to the field as it becomes more dynamic, more impactful, with many real and important applications across many domains in life science, research, and industry. And before we begin, I want to briefly share an overview of what we covered last day in multiple sessions that we had. We had a lot of great speakers, panel discussions, project presentations. So it was a really exciting opportunity to be together for all of us. So first of all, we heard about the Omics logic programs in India. India is one of the most populous countries in the world and is a growing economy that is producing millions of graduates every year. Overall, India has over 1,000 universities with 42,000 colleges and over 11,000 standalone institutions in the country that is catering to over 38 million students. And so what our team is doing is working there since 2019, partnering with institutions in the country and addressing some of the gaps in bioinformatics education by offering research opportunities to students that are working with faculty on various data science projects across many life science domains and building a pathway for students to work in biotechnology, pharma, and bioinformatic industries. And after working with thousands of students from around the country, our team shared lessons learned and offered an important perspective moving forward with some new initiatives that have undergone major changes as a result of this pandemic. One key insight actually came out of the panel discussion where we heard about the challenges and opportunities in bioinformatics in India. So we heard from Dr. Raghav and Professor Radana that offered their insights in academic research as well as government initiatives and urgent need for data science in healthcare and biotechnology industry in India. We also heard from our team in Africa, where Omics Logic training has been launched in 2019. Sub-Saharan Africa is home to more than 1 billion people, half of whom will be under the age of 25 by 2050. So it is a very diverse continent that offers a lot of human and natural resources, and they have the potential to yield inclusive growth and, importantly, eradicate some of the challenges, including poverty and various types of infectious diseases. And so what we heard was about how many of those areas of research are being addressed using bioinformatics. We heard from Dr. Anita Gansa from Ghana, Dr. Vokitoi from Nigeria, Dr. Adrienne Lusa from South Africa, and some of our team members, Mr. Adil Awoye and Mr. Hamza, that have worked with numerous students in this region. Overall, it was an insightful day where we got a global perspective. So I want to thank all of the speakers who have shared their perspective on what was going on in Africa and in India. And here are some of my personal takeaways. So first of all, there is a pressing need to review, annotate, and improve data quality. Both the data that is currently available and some of the future data sets that will be shared. We heard from Dr. Raghav how his group of students and himself are working to make that process more effective and offer a lot of resources for those learning bioinformatics. We heard about the urgent need of training in bioinformatics and data science for those who are working on important challenges in medicine, public health, and other domains. We heard about bridge programs that connect students with authentic research opportunities and with industry careers. And we heard about some of the exciting opportunities for students and researchers to be involved in many fields of research, working together with mentors and leaders in these fields. After that, we heard about some of the exciting developments in the world of bioinformatics research and applications to various challenges like virology.
Yeah, sorry, I had to mute all the participants.
Okay, thank you. Sorry about that. So we heard about some of the speakers that are working on bioinformatics and developing new approaches that are used by thousands of researchers and students. And so here are my takeaways from those talks. First of all, there's a growing complexity of omics data, including single cell sequencing, large-scale genomic whole genome sequencing data, and important questions about the junk non-coding sequences, or the dark part of the genome, where important regulatory elements are being defined. We heard about experimental data that can reveal a snapshot of biological activity, but to see the whole picture, there's the need for integration of multi-omics data, time estimation, and mathematical models that can be simulated to understand what these biological snapshots can tell us about trajectories in time. We also heard about the way this pandemic has spurred active research in many areas, leading to new approaches for genomic surveillance, novel types of vaccines, and even new interventions based on our understanding of viral evolution. And finally, we heard about a new, or maybe an old field called astrobiology. First of all, we heard about how it has contributed to the development of genomic technologies in general and what data sets are available, and heard about some of the novel findings coming from the study of extremophiles that are useful on here on Earth, with many applications in biotechnology. And these talks were accompanied with a panel discussion on the topic of omics data and associated methods of analysis. So we heard about the perspectives on how this field of bioinformatics is playing a major role in various research projects, from Dr. Raul Andino from the University of California, San Francisco, Dr. Sona Vasudevan from Georgetown University, who's also going to be speaking today. We heard about Dr. Leonid Brodsky from the Tauber Bioinformatics Research Center at the University of Haifa, and Dr. Koussoulis from the National Association of Idea Principal Investigators. He also will be speaking today. And finally, we heard from Dr. Laura Harris from Michigan State University. So what these speakers highlighted was the role that the students can play and some opportunities for early career scientists in many areas, including virology, microbiology, biotechnology, and as well as clinical and biomedical research projects to utilize bioinformatics and get involved in many of the projects. So briefly, I want to tell you what is the reason that we are organizing this symposium, and how can we transition from some of the topics that we heard about yesterday and go on to the topic of today's sessions. So the Omics Logic programs, which are operated by Pine Biotech, are designed to provide training in bioinformatics that enable independent research guided by mentors and peer example. It is also to develop a growing community that shares a passion for data-driven research and many applications of citizen science. So it is really a goal of ours to build this community, and part of that process is this symposium. And ultimately, our goal is to enable students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. And so the way that we do this is by offering training programs that provide access to high-performance computing, mentorship, and training in bioinformatics and data science for biological data. The training modules are offered through the Omics Logic portal, where you can register for free and explore asynchronous modules on various types of omics data, exercises in pipeline building, coding in R and Python, as well as offer a view of case studies based on peer-reviewed publications from top research journals. So the link to the portal that you can explore today as well is right here. What these projects are organized into learning paths for oncology, infectious diseases, neuroscience, and agriculture. In each learning path, we use examples that illustrate major trends and novel applications of omics data in particular field. And since the domain of bioinformatics is so dynamic, we are relying on collaborators in all of these fields to navigate updated topics and developments. We work closely with our partners at the Tauber Bioinformatics Research Center, where the team is making many algorithms for data analysis easy to use for non-bioinformaticians using the T-Bioinfo platform, which offers a user-friendly interface for many commonly used methods on phylogenetic, analysis of gene expression, alignment to reference genomes, analysis of structural data, and variant calling. And so this T-Bioinfo platform is also available on this link. Okay, sorry about that. There we go. So one of our programs that we highlighted throughout the symposium is the Research Fellowship, where students can design an independent research project and work in groups with mentors that guide them to complete their research using bioinformatics, relying on publicly available data sets and working with mentors that are provided through our program and through our network. Many of the research fellows have been working for the past several months to complete their projects and share them for the poster session that you see today. You will find the projects that they have submitted organized on this link. So please, Navia and Ojasvi, post the link to the poster presentations, where we are also expecting feedback from the judges, as well as all of the other participants. So you can all go to this page and actually, thank you, Ojasvi, and actually see all of the posters and also contribute your feedback as well as other participants of this symposium. So briefly, let me introduce kind of an overview for the plan for this day. So the main goal of this day is to introduce you to some of the ways that you can continue your journey into the field of bioinformatics, joining degree awarding academic programs, which we will hear about today for bioinformatics, and also thinking about your career in either academic research or industry. So we'll hear from some panelists that will describe what does it take to go from academia to industry and what are some of the opportunities there. So first of all, we will talk about some of the programs. So we will hear from Professor Seema Bhatnagar, who has a history of research in medicinal chemistry and is working with the higher education. And she is skilled in organic synthesis, drug discovery, drug design, protein chemistry, and biochemistry. And she will speak about the program at Amity University. We will also hear from Dr. Sona Vasudevan, who is the director for Systems Medicine program at Georgetown University Medical Center in Washington, D.C. in the US. We will hear from Dr. Philip Compeau, who is an associate teaching professor and assistant department head in the computational biology department at Carnegie Mellon University. He's a passionate educator who has taught a range of STEM courses to a diverse group of students, and he is an experienced administrator who is working on this program at Carnegie Mellon. We'll also we'll hear from Dr. Kasoulas, who is a professor of virology and biotechnology in the Department of Pathobiological Sciences and the director of the Division of Biotechnology and Molecular Medicine, or Biomed. He is also the director of the NIH-funded Center for Experimental Infectious Disease Research and the director of the NIH-funded Louisiana Biomedical Research Network, Molecular and Cell Biology Core, and the president of the National Association of Idea Principal Investigators. We will hear from Dr. Chindo Hicks, who is a program director for a bioinformatics research program with a focus on health and health disparities, including their research and some of the academic awarding degrees that they offer. We will also hear from Dr. Chris Taylor, who is a researcher focused on computational biology and bioinformatics, and especially in the applications of high throughput sequencing for microbiology. We will finally hear a presentation by Dr. Shamir Kader, who is a Senior Director of Advanced Analytics, Data Science, and Bioinformatics at AstraZeneca, and is leading a global team of data scientists, biomedical scientists, statisticians, and software engineers to address some of the pharma-oriented challenges in this field. And finally, we will conclude with a panel that I'm very excited to hear from all of the speakers, where we will hear from Dr. Shamir Kader, Tarun Walia, as well as, Anthony Finlow from Eagle Genomics. And I see that there is a mistake on the slide, but we will correct that. And they will speak about some of the ways that you can get involved as a student, as an early career scientist in this field, in the field of bioinformatics. Where are the different opportunities, and what are some of the challenges between academic and industry approaches to bioinformatics? So finally, all of the poster presentations are going to receive feedback from some of the judges. We have an excellent panel of judges: Dr. Sona Vasudevan, Chris Taylor, Philip Campeau, Julia Panov, G. Poliano Chavez, Abhishek Sengupta, and Dr. Harprika Ur. So they will all provide you with feedback on the posters. Ojasvi and Navya here from our team will be monitoring the chats. So if you have any questions, Ojasvi and Navya, I see there is a question already. And before we jump to our first speaker, I would like to ask Sonalika to launch the poll to kind of get a sense of who is participating today with us. So Sonalika, could you launch the poll?
Yes, Ilya. Thank you so much. So I have 3 questions. My first question is, what is your educational background? Are you a graduate or a postgraduate from PhD or postdoc? What is your designation, student, faculty, researcher, or are you from the administration and management? And what are your research interests? Is it infectious diseases, data science, genomics, transcriptomics, metagenomics, epigenomics, or R or Python for bioinformatics or precision medicine?
Okay, so you launched the poll?
Yes, it's launched, Ilya. I can see that 40% of the participants have voted, and I would be keeping the poll on for more 30 seconds. It would be great if you could all please vote so that we know from what all backgrounds you are.
Okay, and as we wrap that up, Mohit, would you be able to introduce our speaker, Professor Seema Bhatnagar?
Sure, Ilya. Thank you. It's an amazing experience to be a part of this short journey, and looking forward to all the upcoming sessions. And it's an absolute pleasure to introduce Professor Seema Bhatnagar, who is an assistant director at MIT Institute of Biotechnology, MIT University, Noida. Professor Seema Bhatnagar is also an experienced professor with a demonstrated history of research in medicinal chemistry and working in higher educational industry. Professor Bhatnagar has been associated with research and development in the field of drug discovery and working with various government and non-government organizations before her current assignment with
Okay, sorry. It sounds like maybe Mohit had some difficulties in connecting. But Professor Seema, it's great to see you here. And please share your screen so we can all see your presentation.
Thank you, Elian.
Sorry. Am I audible?
Yes,
Yes.
but I think, yes, so Dr. Bhatnagar is sharing her screen already, so we can let her continue with the presentation.
So are my slides visible?
Yes.
Okay.
Thank you. So good evening, everyone from India. I'm Professor Dr. Seema Bhatnagar, Assistant Director at Amity Institute of Biotechnology. And today I'm here to talk about folate receptor, a rational therapeutic target for cancer treatment. This is the area in which I'm working. And I will also be speaking a bit about the programs at Amity as per the session schedule. So cancer, as we know, is becoming a global epidemic, and we are seeing more and more cancer cases. It is estimated that 1 in 5 people will be suffering from cancer in coming years, and this includes 1 in 8 men and 1 in 8 females will also succumb to the disease. So this is something really alarming and something that requires immediate attention. And so that is what is the focus of my talk today. We are also seeing that in middle and medium and low income countries, there will be a steep rise in the cancer cases as predicted by 2040. As compared to the countries where already there are more cases, but in future we will see more cases in countries which are of low and medium income. So India falls in that category, and that's why this is a matter of concern to everyone. If we talk of the cancer mortality, the situation is alarming as well. So if you see the gender divide, males and females equally are equally prone to the disease, and we are seeing a high mortality of the disease both in males and females. And India has a high mortality rate. So historically, if we look at the drugs or the chemotherapeutic agents which were available for treatment of cancer, we see that the first attempts to look at chemotherapeutic agents started somewhere in the late 1940s. And truly the decade between 1960 to 1970 can be called as the decade of adjuvant chemotherapy, where a number of agents were not only discovered, but they were also tested. And many of them were from plant sources, and they are still in use today. However, they have a number of drawbacks, and that is the reason for the growing concern about how we can improve the quality of life of individuals and how we can prevent the disease, or if not preventable, at least treat them and improve the quality of life of cancer patients. The last 20 years from the year 2000 has seen a large number of small molecules that are coming up for targeted cancer therapy. There are a number of targets that have been explored, and small molecules that target these receptors or pathways for cancer have shown a lot of promise. However, if we look in terms of the experience of patients, we don't see much improvement, and according to the World Health Organization, every year in India, at least 10 lakh individuals suffer from the disease. And the spending in a cancer suffering household is at least 36 to 44% higher than as compared to households of similar demographics. Even the cost of treatment has gone up. So what is really wrong? Are we not having drugs? Or if we are having drugs, then why are we not able to reduce the cost? And what is the way to improve the quality of life of individuals who suffer from the disease? So this is something that that needs to be looked into. So all this has given, initially there was, the focus was on traditional medicine, which included radiation, chemotherapy, and surgery. But now this has given way to more targeted therapies, the involvement of genetics of individuals, and also immunotherapy for treatment of the disease. So our group at Amity is involved in study of the folate receptor, and we are looking into new drug delivery options, how we can deliver low concentration of the drug to the cancer cells in a targeted way. And the folate receptor appears as a very attractive target for cancer drug delivery. The reason being that, first of all, it has been newly identified, and there are 3 isoforms of the folate receptor alpha, beta, and gamma. However, the folate receptor alpha has seen a lot of success and a promising therapeutic target in a variety of cancers. Non-malignant tissues express very limited amount of folate receptor alpha, whereas the malignant tissues, and especially solid tumors, the ovarian cancer, kidney, lung, brain, breast cancer, lung cancer, and you name it, all these have overexpression of the folate receptor. Also, what is important is that the folate receptor alpha has high affinity for folic acid, and conjugates of folic acid can be considered as good opportunities to target the cancer cells. And a number of clinical trials are also ongoing in this area. So I will spend some time talking about the accessibility of the cellular uptake through the folate receptor. We know that 80% of the human cancers are derived from epithelial cells, and those receptors that are restricted to the apical regions of the cells can be considered as good targets, because when drugs are administered parenterally, they are not accessible to normal cells. And this is a big advantage, because most of the drugs that were already available, they lack selectivity, especially the chemotherapeutic drugs. They lack selectivity. They show a lot of toxicity, because they not only target the cancer cells, they also target the normal cells. So if you are using a targeted approach, then obviously you will be able to take care of the off-target toxicity, which is seen in most cases. So since receptors are apically expressed, so when the drug is administered parenterally, it is not accessible to the normal cells. However, when the cells become malignant and the tight junctions collapse, in those cases, the folate receptor becomes redistributed on the cell membrane, and that is the reason why it becomes accessible to drug conjugates which may be administered parenterally. So this is a big advantage, that where you can opt for targeted drug delivery. So 3 general strategies have been explored. We have anti-folate receptor antibody approach, the humanized folate receptor alpha antibody drug conjugate approach, and the folic acid SMDC approach. SMDC is the small molecule drug conjugate approach. So if you look at the molecule of folic acid, it comprises of 3 regions. You have the pteridine ring, you have the para-aminobenzoic acid portion of the molecule, and you have the glutamic acid. So it is the pteridine ring which is primarily involved in the binding to the folate receptor. Therefore, this part of the molecule is very much available for functionalization and can be used for drug delivery. So a couple of drugs just as examples. So here we have the antibody approach where farletuzumab has been tested not only in phase 2 but also in phase 3, and what has been seen so far is that it did not meet the primary endpoint of improving progression-free survival in patients. There are also some studies which show the antibody drug conjugate approach, and MIRV is one such molecule, one such agent which has been tested, and it is showing some promise, but still in phase 1 and phase 2. Then we have the folic acid small molecule drug conjugate approach. So the folic acid conjugates can be used to deliver the drug payload to the cancer cells. So, what it involves is that folic acid can be conjugated using cleavable bonds, and these can carry the drug payload to the cancer cell. Once the folic acid binds to the receptor, it is internalized, and the internal pH of the endosome will result in the cleavage of the folic acid from its receptor and also release the drug payload. Now, once the drug payload is released, the folate receptor recycles back to the cell membrane, and the drug is delivered at low concentration to the cancer cells. So that is a very interesting approach, and our work is in that direction. However, we have made some changes which I will be talking about. So vintafolide is one such molecule where they have tested a vinca alkaloid and they have tried to deliver it to cancer cells. This molecule showed a lot of promise. However, it is now out of phase 3 trials. So keeping all this in mind, we looked into the literature, and curcumin, which is a well-known spice which is used in Indian food, and it has a lot of medicinal property. We looked into this molecule. It's a wonder molecule because it has a whole range of activity and shows a lot of promise, but there are some things which have to be taken care of. So it has been associated with antioxidant, anti-inflammatory, anti-cancer, antiviral, antibacterial properties. It is also known to chemosensitize many chemotherapeutic agents such as 5-fluorouracil, doxorubicin, paclitaxel, cisplatin, etc. The only drawback of this molecule is its poor bioavailability and poor absorption. So that is what limits its use in the clinical setting. So keeping in mind the multiple targets that this molecule is known to modulate, we have designed our own small molecule drug conjugate, which comprises of folic acid. There is a spacer unit which connects it to curcumin, and finally there is a cleavable bond that is connecting to an anti-cancer drug, and we have used paclitaxel. We have used acid-sensitive cleavable bonds, which can cleave in the acidic pH of the endosome. So I'm going to share some of the results, preliminary results that we've got with this triple conjugate that we have designed. We have tested our conjugate in the cervical cancer cell lines like HeLa and SIA cell lines. And what we find when we do the MTT assay of these compounds is that our conjugate is working as good as the known drug doxorubicin, which is really important because curcumin might be assisting in its in the drug entering the cancer cell. The cell proliferation assay also, when we compare it with curcumin, doxorubicin, and with untreated cells, we see that it is showing comparable activity at 12 and 24 hours, both in HeLa and SIHA cell lines. In order to look at its ability to enter the cells, because it's a large molecule, is it able to really enter the cells? So we have carried out these studies where we have seen that our molecule is able to enter the cells, and this green fluorescence that you are seeing is of curcumin, which has entered the cancer cells after treatment. We have seen it at 6 hours and 18 hours using not only curcumin but also the conjugate. And our conjugate exhibited better fluorescence as compared to curcumin, which is evident from the figures. We also carried out some in vitro cell migration assay using our conjugate, doxorubicin and curcumin, and with the controls. And what we found is that in case of the untreated cells, whereas there was 100% migration of cells, but when we treated with either of the agents, we could see that cell migration was very much inhibited by using the conjugate specifically. So we've also compared the apoptosis of our, we have carried out the apoptosis assay with our conjugate curcumin and doxorubicin in the same cell lines. And we find that this molecule, this conjugate, in fact, shows the cells are more apoptotic when we treat them with the triple conjugate. So just to summarize what we have got so far is that our conjugate is exhibiting better cytotoxic activity as compared to curcumin or doxorubicin alone. There is 30 to 70% reduction in cell proliferation in both HeLa and SIHA cell lines. In our uptake experiment, we did see that the conjugate could internalize in the cell, and we have seen it visually that more concentration of the conjugate or curcumin, which is could be seen inside the cell, which means that indeed the conjugate was internalized inside the cell. The scratch assay again showed significant reduction in the migration rate as compared to the untreated controls, and the apoptosis assay after treatment with curcumin doxorubicin showed that the apoptosis rate was much higher when we used our conjugate. So all these studies gave us a lot of confidence, and now we are proceeding ahead with more in vivo studies for our conjugate, and we believe that curcumin would be the game changer for this conjugate, because it is assisting in the entry of the drug by chemosensitizing the cell. Of course, more studies are required for it, but our preliminary results indicate that probably curcumin is assisting and the drug is able to enter the cell at low concentration. So if it is able to enter at low concentration, we are able to also target the cell. So we expect that the off-target toxicity will be taken care of, and at low concentration, we are expecting similar results as compared to the known drug. So my next part of my presentation is about the graduate and the postgraduate programs at my department. So we are Amity Institute of Biotechnology at Amity University, and we offer a lot of programs in the area of biotechnology, which includes BTech, BTech Bioinformatics, BSc programs in biotechnology, BSc MSc Dual, BTech Dual, 3-continent programs. And we also offer BSc Biotechnology and B.Ed courses. And for our postgraduate programs also, we are offering the MBA Biotechnology program, MSc Biotechnology, Bioinformatics, MTech Biotechnology, and of course our PhD programs. We also have some certificate programs in artificial intelligence. We have Masters of Technology in Bioinformatics. So it's a big focus in our department, and we have a large number of students who are really interested in all aspects of biotechnology. Our institution also offers many opportunities for international opportunities for students, which include the student exchange programs, which are offered in France, where students can spend a semester abroad and complete their studies in a foreign university. We offer study abroad programs at 4 or 5 locations all over the world, and these are available to all students, UG and PG students at Amity Institute of Biotechnology. We also have a very unique 3-continent program. Now, this program, we have 3 streams in which we run them. The BTech Biotech 3-continent, BSc Medical Biotech 3-continent, and the BSc Honors Biotech 3-continent. So in this, the students complete most of their studies in India, but in the last semester, they travel to UK and US for 3 months each, and finally, they get their degree from Amity University. We are also collaborating with many universities abroad, and we are offering international collaborative programs in BTech Bioinformatics, BTech Biotechnology International. Again, it is a 2+2 program with Indiana University, and the BSc Honors Biotechnology program is with the University of Queensland, Australia. It's a 1+2 program. So students have a plethora of opportunities if they join the department, and truly we believe in creating global citizens at Amity, and our students have always made us proud. So and we hope to continue the same in future. So thank you very much. Thank you for your patient hearing. If there are any questions, I am ready to answer them.
Thank you very much for this presentation. We do have a few brief questions here. One of them are there any specific techniques required to synthesize these kinds of molecules? He means conjugate.
Yes, so conjugation chemistry would all involve chemical synthesis of the conjugates, but yes, it's very important to characterize whether conjugation really took place or not. So, of course, you would be using IR, NMR, and you have to use, since it's a large molecule, so you may have to go for MALDI-TOF to know the molecular weight of the molecule. So all that is very important, because just carrying out the synthesis does not mean that the conjugate got made. You have to characterize it.
And a lot of people are asking about how they would join your department or your programs. So if you could please post a link to the department page to learn about the program a little bit more.
Yes, I will just do that in the chat box. I will do that.
Thank you very much. And Sonalika, we'll pass it on to you to introduce the next speaker. Thank you very much, Dr. Bhatnagar.
Thank you. Thank you, Ilya.
Thank you, ma'am. Thank you.
Thank you so much, Professor Seema Bhatnagar. Now, I would like to welcome Dr. Sona Vasudevan. Dr. Sona Vasudevan interests spans the areas of structural biology, structural bioinformatics, homology modeling, protein ligand and antibody-antigen interactions in RNA biology. Her research interests include comparative genomics, protein classification, protein homology modeling, and molecular dynamics simulation to look at the effect of single nucleotide polymorphisms in breast cancer, evolutionary analysis, systems biology, and translational informatics. Dr. Sona Vasudevan would be talking about the systems medicine education at Georgetown University. Over to you, Dr. Sona.
Thank you, Sonalika, for that wonderful introduction. Thank you, Ilya, for the amazing opportunity to be part of this symposium. I've enjoyed it very thoroughly. So good morning, good afternoon, good evening, good night. I know there are lots of participants from different parts of this world. So I'm just going to spend the next 15-20 minutes to tell you about, let me share my screen. Okay, I hope you can see my slides here.
Yes, ma'am, we can see your screen. Thank you.
Yes, so I'm going to spend the next 15-20 minutes to tell you about an educational program that we have developed at Georgetown University, which is Masters in Systems Medicine. Of course, before I delve into the details of the program, I want to let you all know, those of you who have not visited Washington, D.C., you're welcome to visit because it's actually a very beautiful place out here, especially during the spring. And Georgetown University, of course, has expert faculty and a beautiful environment as well. So before I dwell into the program itself, I'm not sure how many of you have heard of the term systems medicine, because this is a very new term, but I think it's becoming more and more secondhand to those of us who actually practice informatics. So for those of you who have not heard of this, I just want to give you a brief definition of what systems medicine is. This is a very traditional definition developed by the European Systems Medicine Association. So it's a novel approach to medicine. It is the first step on the path to personalized medicine. It's based on computer models in which vast amounts of clinical data and are used to analyze health of individuals and patients. It promises to further our understanding of disease mechanisms, to boost the efficacy of treatments, and of course, cut costs of healthcare. In a nutshell, what systems medicine means is it's one aspect of personalized medicine. And we know that even with COVID, we're all experiencing there's no two individuals exhibit the same symptoms. We have a wide variety of symptoms affecting wide variety of age groups and a wide variety of outcomes. This is the practical definition of systems medicine. It is a multidisciplinary field that looks at an individual as a whole. It uses systems biology and computational sciences and omics technologies to help solve biomedical problems. And it is the future of medicine. And I'm sure you all will agree with me with the explosion of data out there. There is no going away from looking at data and putting things together. We had lots of interesting talks yesterday in the symposium about different integration techniques of all of this omic data and what the dire need for educating informaticians. And Dr. Bhatnagar at MIT just talked about amazing programs to expand this field of informatics, and that is extremely welcome. So in summary, what systems medicine is all about is the realization that one size does not fit all. Two patients with the same symptoms, same findings, same disease are given the same drug, but each patient exhibits different outcomes, different reactions to these drugs. And why does this happen? Why does drug response vary? This is what gives us the multidimensional problem to solve, which is at hand right now. So it all boils down to individual variations between two different individual patients. And these individual variations stem from genetic differences between individuals. It could be as simple as one single nucleotide polymorphism. I don't have to emphasize to this audience that the majority of the diseases that we see is actually caused by single nucleotide changes. Sickle cell anemia is just one single mutation. And we're talking about a lot of variants right now. So it's all single nucleotide variations. So there are lots of variations that exist among human population. And there is ethnicity differences, age, genetic factors, diseases, drug-drug interactions, environment, nutrition. So these are all some of the major factors that lead to different drug responses among the different individuals. And why are we talking about this right now? I don't have to, again, emphasize to this audience. So we are at the midst of an era of big biological data, and we so happen to be in the middle of a pandemic as well. And a revolution in medical practice is happening as we speak, with all of this data coming from different omic technologies. So it all started here with the Human Genome Project more than 2 decades ago. Successfully, we were able to map the total number of human genes, sequenced human genes. Of course, we are still continuing to sequence the human genome to get the most accurate picture of it. But today we stand here knowing that we have the total number of genes in the human system is about 20,000 to 21,000. So this initiated a whole cascade of different avenues to pursue. And of course, when the good news about the human genome sequencing project was completed, all of us, including myself, celebrated, thinking that this is the beginning of end of a cure for all diseases. But here we stand after 2 decades. We are still not there yet. We have made lots of progress, but we're still not there yet because of the complexity that exists in the human genome itself. Of course, omics space, all of you are familiar with lots and lots of different omics, and we are in the middle of an omic era. So omics space is actually the pillar of systems medicine. So we have lots of different layers of omic: genomics, transcriptomics, proteomics, you name it. And genomics is being used to integrate pathways, mutated genes, drug-drug interactions for various diseases. To add to all of this complexity, we are now dealing with another omics, which is the microbiome, with all of these individual organisms that we all carry alongside our human genome and human cells as well. So these are all the different omics where we have a huge amount of data coming out of all of this. And now the challenge that faces us is, how do we look at and integrate all of this and come up with an understanding of diseases and understanding of human biology. So genomics has become an universal diagnostic tool in clinics right now. And to understand the molecular mechanisms of diseases, I can't even think any other way of looking at a disease right now. Even when I did my PhD about 2 decades ago, all we did was study one gene, one protein, and the implication of one gene and the mutations in it in a disease. But here we can't do that anymore, because we have come to an understanding that diseases are defined more complex. Of course, I'm not talking about the monogenic diseases. Majority of the diseases are more complex than that. So this has led us to look at genomics, and now we are currently using that as a diagnostic tool. So systems medicine is already in clinics, and it's surfacing in various different ways. Big data, using large data sets to learn about general population, clinical decisions, and all of those things. Of course, reproductive endocrinology, testing embryos, so potentially fatal conditions. We are here doing all of this today. Genetic testing is available widely, and we're able to catch a lot of metabolic disorders in newborns. And tumor profiling, using genetics to tailor cancer treatments by tracking mutations. And pharmacogenomics, assessing individuals' prospective reaction to a drug based on their genetic specific makeup. So we are already seeing systems medicine in a day-to-day practice of what we are doing. And here I stand today to let you know that there is a revolution in medical practice, and we're all about efforts about disease prevention and promoting health. That is our goal. So all of this data and all of this has made us realize that we are learning a new language. And it has led us to realize that we actually need new educational programs to learn this new language. And this education, in my opinion, has to start from schools, high schools or undergrads. And so Georgetown University, we realized this about a decade ago. And also, of course, all of you will agree, an investment in knowledge pays the best interest, right? So that led us to create a dual master's degree program in systems medicine at Georgetown University. This was launched in 2011. And the program, of course, I'm not going to go in depth about the curriculum itself. There may not be much time, but I'm happy to talk about it and send you some links that, and you could go and look at the details of the curriculum. The curriculum for this master's program mainly was intended for students in medical school who have completed years of medical curriculum, because these are physicians that are going to be using this data for treating you and me. So this program was intended to generate a new type of physician, one with a grasp of modern computational sciences and omic technologies, genomics, proteomics, metabolomics, and translational sciences, etc., so that they can embrace this into their practice of medicine when they are looking at that. And of course, more importantly, to understand the systems approach to medicine, because one size, as I mentioned, one size does not fit all, and it is not about one gene, one protein. It is about a whole systems that actually we have to look at in order to understand a disease. So after a successful launch of a dual degree program, we came to the realization that this language has to be taught to all students, no matter what their career paths might be. So that led us to create another master's program, which is a freestanding master's program in 2016. And it was intended for any student who's interested in understanding biomedical, any discipline in my biomedical sciences. This is again to generate a new type of scientist, new type of physician, new type of any healthcare individual who can also learn this language to embrace that we can. So again, more importantly, to understand the systems approach to medicine and research. So we launched those two programs, and I'm not going to go deep into the curriculum itself, but just to give you a flavor, students have to complete 30 credits of course requirements. That includes core courses that each student is actually trained in, that include biostatistics, biomedical informatics, critically reading scientific literature, which is becoming actually very, very important. Unless you understand how to read a scientific paper, we're not going to be able to understand what is out there, and to critically review different techniques and technologies to make amends and improvements to all of those. And to be able to understand how to analyze clinical patient data that's out there. We have huge amount of data that's available in different resources. And of course, as I mentioned, microbiome is becoming extremely important, and what are the different organisms that are up and down regulated in different situations? What is a normal microbiota? So we have courses that teach the students how to look at microbiome, and what are the different aspects to genomics, and how do you take data from bench to bedside, and how do you translate all of the data and the experimental data that is out there, and looking at metabolomics. Everything is about metabolomics. Any reaction in our body is going to give you metabolites. So this is the a picture of the courses that students actually learn. And of course, in addition to the core courses, students actually end up taking elective courses based on their interests and based on their career paths. So we have several different courses that span from looking at drug design, three-dimensional structures, to looking at how do you think in systems, right? And also how do you create policies, and how do you look at all of this in a lab, and how do you learn all of this? So this is, of course, the details of all of these courses. You can find it on our website. And Students are allowed to take courses across the entire different departments at Georgetown. They can learn about epidemiology, or they can learn about diets, they can learn about supplements, and all of those things. The most important part of the curriculum is a capstone project. So students are mandated to do good portion of their time they spend doing research. So you can learn as much as you want in a class setting, listening to lectures, but unless you know how to learn to apply these in a research setting, it's not going to be a practical use, and we won't be educating the students to use of all of these different techniques and resources and tools that they learn in a class setting. So capstone is a very integral part of the program. And students spend more than 240 hours doing research, and the research culminates in a poster presentation, and majority of the students end up publishing, being part of a publication. So which as they learn how to write a paper, they publish a paper, and they know how to apply all of the tools that they're learning. So research is an integral part of the program. And students have a variety of options to go and do their internships. We have several mentors. This is just only a flavor of the institutions in and around Georgetown where students actually go and do their mentoring. Of course, in addition to all of this, students have mentors in industry and other universities as well. So we have a wide network of collaborations across different universities and industry settings. So the question is, what are the skill sets that these students actually get out of this program? And if you are out there looking to hire, these are all the skill sets that they get. So they understand what is big data. So what it means to actually deal with humongous amount of data. So they also understand what sequencing means. They understand how to carry out next-gen sequence analysis, RNA-seq analysis, microbiome data analysis. So they are exposed to how to do use the data and meaningfully extract knowledge from these data. We also teach biochemistry, which I think is fundamental, whether you're a researcher or whether you are a physician or anything, you have to understand the biochemistry. But the way we teach biochemistry is based on cases. It is about patients' cases. So they understand that a patient walks into the clinic, has these symptoms, and they understand how to troubleshoot to figure out which gene is going havoc in them, what mutations they have, which pathways it's implicated, so that they have an understanding of the disease from a systems perspective. And thanks to Pine Biotech and Ilya, we have been able to launch a course on machine learning and AI, and I'm very excited about this. So students learn machine learning as well aspects, and they also learn about healthcare in terms of electronic health records, electronic medical records. How do you mine them? How do you get value? How do you take basic research from bench to bedside? And they also, of course, learn how to read an article, scientific article, which is actually very, very important for you to be able to distinguish between good papers and critically thinking and analyzing the results of a publication. They also learn how to mine patient data, how to use patient data, how do you understand the comps of all of these patient data. Last but not the least, they also learn to get their hands a little bit wet to understand how do you generate good DNA or RNA that can be sent for sequencing, and what are the caveats in them. So how do you understand between good data, good sequencing data, and bad sequencing data. So we expose them to the nanopore sequencer. So they learn how to actually hold the sequencer and do the sequencing. So these are all the skill sets that the students actually learn from the program. And there is something for everyone. So those students who actually come to us for the MS program, they of course go and become physicians. And those students who actually do the MS program go on to medical school, nursing school, BA or GO schools, or go into PhD programs, or work in industries, academic settings, and of course, healthcare and doctor's offices. So there is something for everyone in the program. So we are all about skill sets. And so they develop lots of skill sets that they can apply in different industries and different job settings. So the question that's always asked is, where are the graduates now? So the graduates are all over the place here. Some of them are in medical schools, PhD programs, nursing schools, and of course, several of them are working at several different universities, NIH, FDA, and all of those. So, so the students get placed pretty well. And of course, why Georgetown? And I'm proud to say that the program is the first in the nation. And of course, we have outstanding faculty and we have an outstanding team to help us. Of course, it all boils down to whether we have scholarships that we support the students. Yes, we do have several different scholarships that students can apply to. So we have an endowment of $100,000 that was generously provided by Robert Stephen Ledley endowment scholarships, and we also have lots of different biomedical graduate scholarships. Of course, all of the details are, okay, you can find it in the website, and I'll post the link to our website in a second. Of course, it's not all about academics. Students have a very good life. We go out for lunches, we party, we celebrate different things, and we have a social life as well. And there's a wonderful academic life as well. Students attend conferences, they present their work, they are taken to field trips to the NIH, see the sequencing facilities, and you can see all of them are smiling, so the program does treat them well. And so they do have, in addition to their academic life, they do have a good social life as well. So this is the website to the program if you want to learn more. And I just want to actually leave a few minutes for questions so that I can elaborate on anything else that you may need from this. And I want to thank you for your attention. And if I'm happy to take any questions you may have.
Okay, thank you very much, Dr. Vasudevan. We do have a question. Do you have an opinion on the advancement of AlphaFold that will change the scene in structural biology field in the near future?
That's actually an outstanding question. I am a structural biologist. I am a structural biologist by training myself, and yes, it is going to completely revolutionize the way we are going to do structural biology. And as you know that already, people are using AI and machine learning. And in fact, we have a structural biology team at Georgetown University who have actually used the concepts to come up with some, therapeutics for COVID-19. And the answer is yes. I've been waiting for this. And the answer is yes. This is going to move the field very fast. So that we'll be able to design drugs in a much more systematic manner as well. What we could not do before, we're able to do because we don't have to depend on the experimental sciences. But of course, there are lots of caveats still. We are not there yet, but we are getting there. And I think this is definitely going to revolutionize. I'm very excited. I'm actually learning and using it myself. So the answer is yes.
Okay, thank you very much. So in your opinion, how do you see someone with a master's degree in molecular biology and genetics fit into systems medicine?
Oh, actually, I would definitely welcome. In fact, I would love to have students who have the background in biology and genetics to be able to contribute to informatics. As you know, informatics is actually an application field. We do depend on experimental biologists and the knowledge from experimental biologists to make sense of all of this data that is coming out. And in fact, most of my students who come, actually, I require that they have a biology background. And if you have a molecular biology background and genetics, that's an added, it'll be an asset to have such expertise in the program. And you will actually gain much more than just a person without a little bit of biology background.
And so the final question is two-part question. So do you have any international students that join your program? And if somebody like that wants to apply, how are they going to handle the registration and fees? It seems like the program is very expensive.
Well, this is actually some of my heart bleeds because I do want a lot of international students. And what limits is the expense? Yes, the first part of the question, the answer is yes, I do have international students. I have graduated almost about 7 or 8 international students in the program since its inception in 2016. And I know that expense is a big problem, and I'm trying to find funds. There are merit-based scholarships that the program provides, and if you have a merit-based, if you qualify for that, you definitely, of course, it's not a lot of money. So that is something that I'm actively pursuing to figure out how to generate revenue so that I can create more scholarships for international students. So we do have a lot of international students actually at Georgetown, and personally, I do. In fact, I have a couple of international students who are going to be starting tomorrow for this fall. Yes.
Okay, so thank you very much. We do have a lot of other questions here, but we're not going to have time to answer all of them. There is a link that maybe you could share on the chat as well for anybody who wants to learn more. And you can also go to our symposium page. We have links to all of the participants, or sorry, all of the speakers, all of the links you can find there. Okay, so Monica, over to you. Thank you very much, Dr. Vasudevan. Great to hear about all of this.
Yeah, thank you so much. Thank you all for your patience.
Thank you so much, Dr. Sona Vasudevan. Now, I would like to welcome Dr. Philip Campeau. He would be talking on undergraduate and high school computational biology education at Carnegie Mellon University. Dr. Philip is an associate teaching professor and assistant department head in the computational biology department at Carnegie Mellon University. He is a passionate educator who has taught a range of STEM courses to a diverse collection of student groups, an experienced administrator who works to make the student the central focus of our educational programs, and a leader of several successful projects in open online education. Over to you, Dr. Philip.
Okay, thank you so much for the kind introduction. I'm going to share my screen. And you can find information about our department at cbd.cmu.edu. So we have a nice short URL. And that's going to have information about the programs that I'm talking about today, as well as all of our educational programs. So we're in Pittsburgh, Pennsylvania, in the US. We have a PhD program that's joint with University of Pittsburgh, which is actually just down the street from our university. It's kind of rare to have two universities that actually are directly adjacent to each other. We also have 2 master's programs, one in computational biology and another in automated science. So we have opened a laboratory that is totally robotically driven, where students can learn about how to not just program the robot to perform experiments in biology, but also to train the robot to make decisions about what future experiments to run based off of the results of experiments. So you have this closed loop experimentation system. But what I was going to talk about today is 2 projects that I have essentially created, one and serve as a director of. One is our undergraduate degree in computational biology, and another is a high school program that I founded with Josh Kangas that educates students over the summer within the field of computational biology, and that program makes us somewhat unique. So I'm very proud of both of these programs. I know that others may talk about more graduate education, but I wanted to share the type of thing that we do with both undergraduate and high school students. If you're interested in our other programs, I would encourage you to go to our website and check them out, because we have wonderful programs for every variety of interest. So in our undergraduate program, we accept students directly to colleges within Carnegie Mellon. So at most American universities, you just apply to the university. And then when you arrive, you decide on what your major is going to be. And everybody can decide on whatever major. And then that means that in many places, computer science has like thousands of people at a time. And so it makes it very difficult to have any sort of personal relationships or this type of thing, because you have just huge classrooms and you're just getting students through. We're a little bit different in that students apply to schools based off of their interest. And so our department is one of 7 departments within the School of Computer Science. The others are things like human-computer interaction, machine learning, and so on. And we have a very, very selective school. So it's one of the most selective institutions in the US. The acceptance rate is only, unfortunately, about 5%. That means that we don't get to see as many talented students as we probably could. We're one of 4 majors that students can choose from once they're accepted, the others being computer science, artificial intelligence, and human-computer interaction. And all the 4 of those majors, students are trained very, very computationally first. So there are plenty of bioinformatics majors out there already. Most of them are aimed towards biologists. And I think what differentiates us is that we are training computational scientists first and foremost. You know, we keep hearing about the computational and big data revolution in biology. And so certainly our students receive a great deal of biological training. We have an entire core of study that's devoted to biological foundational coursework. But we feel that it's just as important to train them as computational scientists alongside some of the smartest undergraduate minds in computer science in the world. And then we have an entire core of computational biology coursework that sits at the intersection of all this math and computer science and biology and physics and chem that students take. And what's nice about this is that those core CS math requirements are the same that computer science majors get. So we know that our students are being put through a very, very rigorous kind of world famous training process at the undergraduate level. One of the cool things that this means that we can do is our students come in and they take a mathematics course that's usually, even by math majors, only taken as juniors or seniors, where they prove theorems and learn about discrete mathematics. Our students do that in their first semester. We know they come in as very, very talented programmers, or if they're not, they become very talented programmers quickly. And so in our second semester, we teach students an entire course devoted to what we call Great Ideas in Comp Bio. So that's our first cohort that we had. And if you search for that, it should be easy to find. I've got an entire website about the course. It contains slides and descriptions of some of our student projects that they did, as well as kind of the philosophy of the course and how we've set it up. We have a collection of course aims. I like talking about this course just a little bit because I founded it with Carl Kingsford and I've taught it for 3 years, where the central aim is to show students what has made biology a computational discipline. It's inherently a data-driven computational discipline in 2021. And so we tell the story, so to speak, of what has happened to make that the case. So throughout this, we find that there's these sub-aims of, for example, you have fundamental algorithmic topics like dynamic programming that comes up several times throughout the course. We take this big, broad view of computational biology to find really cool ideas outside of standard topics. I'll mention that briefly here in a second. We profile topics that have actually been so important that they've gone off and informed things that are outside of biology and computer science. Things like how BLAST works, really, for database search, or things like neural networks. We also point students towards primary research. So at any moment of the course, I always show students the actual research papers that this came from, whether it comes from 1970 or whether it comes from just a few years ago. Our students do group projects, and we also plug in world-leading researchers to give guest lectures to our students as well, so they can see that this is very much a living and breathing discipline. When we teach these first-year students, the first half of the course is devoted to these classic topics in genomics, things like de Bruijn graphs for genome assembly, things like global and local sequence alignment, Smith-Waterman algorithm, read mapping. So we explain how the Burrows-Wheeler transform works and how that's applied to read mapping. Again, this is the sort of thing that you would typically see in maybe a PhD level class or maybe a final semester undergrad course, but we're able to teach it to first-year undergraduates. And then in the second half of the course, we take this big broader view of what computational biology is. And we cover things within systems biology. We talk about the foundations of neural networks, as well as, explore how this has affected this really neat algorithm called the Elan-Cash-Tan algorithm in systems biology. We also talk about algorithms in nature. We talk about spatial and evolutionary game theory, Turing patterns, things that you might not typically see in a 1 year or 1 semester overview course, pretty much because they're beautiful ideas that have led to a lot of other cool work, even if they aren't, at the forefront of what is going on in computational biology today. One of the other cool things about our undergrad degree is that we guarantee at least 1 semester of research to all of our majors. That goes back to our point about going to a university and finding yourself one of 1,000 other majors in a certain subject, and it being a battle to be able to have any sort of research experience. We only have about 20 majors per year. And so that means that we can pair our faculty with all students who want to do research. And we do this because it's what our students told us they wanted. So when we met with prospective students, they kept saying over and over again, I really want to come here so I can do research. What can you do? And so we work with our faculty to guarantee that. We also connect our students downstream to opportunities outside of their degree. We know that our degree is very expensive, to the point that was just raised, and we want to do everything that we can to help students of all of our programs get connected to opportunities in industry, often hearing from what our alums are doing. So this is at one of our events, and we hold these virtually as well. We send resume books of students to alums and contacts in industry and so on. And we also try to have fun with our students too. So even when it's cold in Pittsburgh in the wintertime, we find something to do with them going snow tubing. So that's a kind of a big picture about what we do with our undergrad degree. And our undergraduate degree has only recently graduated its first students who had transferred in from other majors. Our first main cohort, because we've started 4 years ago, our first main cohort is going to be graduating this spring. So it'll be exciting to see all the sorts of things that they head off to do. Although we know they're interested in medical careers, whether or not that means within medical school or not, or going off to industry, whether that's biotech, pharma, or a traditional tech firm. So that's one of the programs that I wanted to discuss. And the other is our high school program in computational biology. That's one of a suite of programs that is offered by Carnegie Mellon. So I think there are 11 high school programs that we have over the summer that are all taught by professors, and that allow students to come to campus under normal circumstances and have kind of a student experience on campus. Our program runs for 3 weeks. Some of the programs run for 6 weeks, where students are in the classroom full-time throughout the day, or working on something, for 8 hours a day for every weekday. And so we have this very intensive, full-time program for a short period of time with rising seniors in high school. So 16, 17-year-old students. We told students, when we started, just as we communicate our goals for my first year undergrad course, we communicate our goals very, very upfront for our high school program too. So we tell students the first and foremost thing that we want to do is have fun. What's wonderful about this, and I'll tie this to point number 5, is that we don't assign grades. There's no metric of success in our program other than just learning. There's no deliverables. Students complete a short project, but in terms of the core content that they're learning, the goal of the program is solely to learn and to have fun doing so. So that means we can really challenge students too, because there are no stakes. We don't have students climbing over each other to try and get a higher grade so they can move on and get into the next school. It's not something that we have to worry about, frankly. As part of this, students usually will learn a new programming language. So we currently use Go. We may change that in the future, but we use a language that's kind of outside of what students typically do, so that students don't come in and say, well, I already know how to program in Python, because that's way students will learn something new regardless. We want them to appreciate that computation is everywhere in biology, that biology is very much a computational discipline in 2021. And we want students to write their own code. So kind of the full cycle of how our program works is that we present students with some biological problem, We explain to them how we're going to solve it computationally. We then show how you might do this by breaking it up into smaller pieces, but the students solve all of those pieces and then put them back together into a very large piece of code that they can then apply to some real biological data set. Those real biological data sets are taken from either the microbiome of the 3 rivers in Pittsburgh. So we have 2 rivers that flow together into a third, and the 2 rivers are very different from each other. They flow through different sorts of environments. One goes by a lot of steel mills, the other is much more rural. And so we captured on a boat, we went out and got water samples with our students from different seasons, and we got a lot of data sets, metagenomics data sets. But we also used the opportunity of the pandemic to look at publicly available genome data sets for SARS-CoV-2 as well. So, but our main topic of focus is in microbiology. And we communicate to students how our classroom works. So just as we communicate upfront our goals, we tell students why we've set up the program in the way that we have. So we talk to them about active learning and about how if we can get our students to do as much in the classroom as possible, as opposed to just listening to me drone on, and receiving information, that regardless of their background, they will see a marked increase in what their outcome is, so in their ability. And that often this doesn't feel like the experience of learning, and other studies have shown that even though students are objectively learning more via active learning, they feel like they're learning less. And we tell students that that feeling of internal struggle is what learning feels like. We use this sort of research, educational research, to inform a transition to our online classroom. So over the past 2 years, we've had to run this program remotely to students across the country. And we use best practices for this. One of the things that we do, partly based off of student input, is we now have students solve problems in teams of 4 or 5. So at most points of the program, students are working together to solve some coding challenge, and they're in small groups doing this, and they can code all in one window that's shared through something called Atom. So they can all see the same coding window, and then they can all run the code together. So this creates this really nice kind of group dynamic where students have input from teaching assistants and input from us, the instructors, as we're roaming around, but they're all working together constantly, so that even though they're online, they're able to really do something all the time in the program. So what is it that we cover? Remember, I mentioned our 3 rivers. We ask, how is it that the bacteria that are present in these rivers change over time? How are they different from one river to the other? How is seasonality important to this? And we use this to come up with simple metrics that are the metrics that are used by biologists, but very easy to explain metrics for quantifying the diversity of a single bacterial sample or comparing 2 bacterial samples, and sprinkle a little machine learning magic on this to produce a plot like this one that shows that seasonality is more important than location for determining bacterial diversity in the 3 rivers. In the second module of the course, and so I've put all the lines of code that students write or that we have as our official solutions. And it shows, I think it's to show how much code that students will write, because typically when students are solving some coding challenge, we don't continue until all the groups are finished. We were worried about whether or not that would be possible, given that many of our students have never programmed until like 2 months before our program, when we send them some preparatory materials. But then all of our groups pretty much solve every single one of our problems. So when we say total number of lines of code, this is, students are writing this much code and using it to solve biological examples. So this module is about, comparing genes, whether it's a database search, and we talk about BLAST. We don't code the entire BLAST algorithm, but we discuss how it works. But what we do code are things like Smith-Waterman sequence alignment, and the students all code it themselves, and then they apply it to something like comparing SARS-CoV to SARS-CoV-2, so they can actually see something that we hear about in the news, which, for example, which is that the variation of SARS-CoV is, isolated to, different points. You have a variable variation throughout the genome. So they can see that based off of the code that they produced. We even write a genome assembler with our students. which is our largest coding project. They're coding as fast as they can go. And we apply it to both simulated and real data to show that on their simulated data that has, some error rate. Our assembler, this is a mummer plot on the left, our assembler does really good job at assembling the SARS-CoV-2 genome. It does an okay job on real data when we cut it loose on real data. And we use this as an opportunity to show that there's pretty much always improvements that we can make, even after we write 1,400 lines of code. If we really wanted a real genome assembler that would truly work on a viral data set, it might need to be 10 times as long as that. And if we were going to apply it to bacteria, it might need to be 100 times as long. But once we can assemble a genome, we can find the genes in it. So we have a short module for students to find genes, and we use this to build a simple annotation of something like a viral genome, as well as to compare our gene finder against an annotation for a larger organism. And students, even with a simple approach, are able to reconstruct like 80% of the genes in, say, a sequenced annotated bacterium. And then in the final module of our class, we produce evolutionary trees, and we apply this to a variety of different contexts. So we look at, for example, coronavirus genomes taken from patients in different countries to understand the initial spread of the coronavirus around the world. We also have students implement a simple version of Clustal, so that this is the actual alignment that came from students' code, so that they can start looking through this, throughout the course of the pandemic, and not just identifying the variants, but defining the variants themselves. And then we compare the student definition against, say, the CDC's definition. What is it that makes an alpha variant? What is it that makes a delta variant? And then we can allow students to classify each of our genomes in some database, say the UK patient genome database, to classify each patient genome according to which variant it applies to. And this is, we just do, normal alpha and delta, but even with this simple classifier that we build, we can see the percentages of the alpha variant go up, very highly in early 2021, and then it's completely replaced with the delta variant. So students, again, have this feeling that they're really doing the research themselves. There's nothing that the real biological researchers did that they couldn't have done themselves now that they have this knowledge. Finally, I point out, we have very, very strong teaching evaluations for our program, but there have been some qualitative feedback that I love to profile here because it reinforces our original goals. Most students, when they learn to program, they learn about it in the context of math. When they think about computer science, they're thinking about Google or Facebook or Amazon. And it's unfortunate that even though this computational revolution has happened, our students, even very, very strong students, many of whom come from very privileged high school backgrounds, have no idea that programming can be used to solve very important problems in biology. And that, many of these problems are very new. For example, with respect to the pandemic, they love the collaboration that we have. They really appreciate the challenge. One student has called it grueling this year, but then said something very positive. So if we're able to balance extreme challenge without frustrating or stressing out our students, then we really know that we're doing our job. And then I always give my favorite quote is from Nia, who was interviewed by a blog to discuss her experience, not just in our program, but with the pre-college program at CMU as a whole. And it was wonderful to see that something that we don't tell our students is that we pair them up based off of their backgrounds, based off of their geography, making sure that all of our groups are kind of ethnically and gender diverse. And we didn't tell our students this, but she recognized, hey, I know that they created our groups to be as diverse as possible, often with different backgrounds, strong biologists or strong computational students. And that's what made the teams work so well. So she was a wonderful student, I should say, but I always try to end with her quote. I'm happy to answer any questions. I don't know where we are on time, but thank you for listening.
Thank you very much for this presentation. We are just very short on time, in fact, 1 minute over time, but it was great to hear about this program. We do have just maybe a couple of questions that we can fit in. The first one is, do you accept high school students from different states, for example, the state of Louisiana? And if yes, do you have a summer program that is online or face-to-face? And if you have a link for that?
Yeah, so we accept students from around the world when we're in person, and we're hoping to be in person next summer. So you can find, if you go to our department website, or there's a pre-college Carnegie Mellon. If you just Google pre-college Carnegie Mellon, you'll find all of the programs. So it may be that you find yourself interested in something else, and those are easy to find.
All right, thank you very much. And it was great to hear the presentation. We're hoping to learn more from the links that you provided. Thank you.
Thank you so much, Dr. Philip, for such a great talk. And I've received comments in my personal chat box that people really liked the talk, and it was very inspiring for some of them as well. Now I would like to welcome Dr. Gus Casolas. He's a professor of virology and biotechnology in the Department of Pathobiological Sciences and director of the Division of Biotechnology and Molecular Medicine. He is the director of the NIH-funded Center in Experimental Infectious Disease Research and the director of NIH-funded Louisiana Biomedical Research Network Molecular and Cell Biology Core. Dr. Gus would be talking on data science for biological data, a message for the National Association of IDEA Principal Investigators. Over to you, Dr. Gus.
Hopefully you can see my screen.
Yes, Dr. Gus, we can see your screen.
Okay, well, I guess good morning or good afternoon, depending where you are. Thank you for the introduction. I'm very pleased to participate in this important symposium. It was exciting to see the opportunities for the elite universities at Georgetown and also at CMU, very exciting programs. I am the president of the National Association of IDEA Principal Investigators, and this is a 503c not-for-profit corporation that is a think tank that helps NIH, and specifically the National Institute of General Medical Sciences, to sort of socialize and support biomedical research within 23 states and Puerto Rico. And those 23 states in Puerto Rico, representing practically half of the country, are those states that do not receive high levels of NIH funding, and NIH is concerned about that and has created this program to support biomedical research, and specifically data science and bioinformatics at the undergraduate and graduate level, with a focus on primarily undergraduate institutions. So NEBI aims to, as I said, be a think tank and advise NIH on programs and how to really support biomedical research. And it fosters interactions, promotes resources sharing, enhances the visibility of multiple programs I will tell you a little bit about, and develops consensus priorities, disseminates best practices, and identifies opportunities and develop strategies for excelling in biomedical research. This is the leadership of the NEPI. I serve as the president, and we have an executive committee with Dr. Doug Wright from Kansas, Dr. Ian Mang from the University of New England, Dr. Scott Seville from Wyoming, and Dr. Julia Oxford, the secretary from Boise State. The NEPI website, I think, is part of my what I have disclosed here, and you could look at the NEPI site for all this information about what NEPI is doing. So again, the mission of NEPI is to provide leadership and communication for the development and promotion, improvement of biomedical research. And as already told you, a lot of the goals is to really foster interaction, support all our constituents and stakeholders throughout those programs. So a little bit briefly about the history of the IDEA program. The National Institutes of Health developed the Institutional Development Award, or IDEA program. It was established in 1993 to broaden the geographic distribution of NIH funding for biomedical and behavioral research. The program fosters health-related research and enhances the competitiveness by investing in institutions located in states in which the aggregate success rate for applications to NIH has historically been low. So it is to a large extent manpower development, socializing support for biomedical research, and supporting in much greater numbers people that could pursue biomedical research. The program also serves unique populations such as rural and medically undeserved communities in these states. So this is just a brief idea about the funding. The current funding is approximately 400 million. That's distributed among different programs. It started much lower in 93 with approximately 30 million and has been rising, hopefully attending up to 1% of the budget. And as you can see, 1% of the NIH budget that is actually distributed for these programs, competitive programs in 23 states and Puerto Rico. And these programs have been fundamental because they increase competitiveness within the states and leverages them to be able to compete with categorical traditional funding by NIH. And a number of our states, because of this funding, have been able to compete and get significant funding by competing with much more developed institutions and states. So the 5 IDEA funding programs is the Centers for Biomedical Research Excellence, the SCOBRIs. This program is really targeting to graduate individual investigators with R1 funding from NIH, and has been fantastically successful, having investigators reaching the first R1 at a younger age in comparison to the average across the states. And also promoting excellence of biomedical research within specific institutions. The IMBRE Awards, for which one I'm the principal investigator, is a different type of award, one per state. There are 24 active awards, and those have a targeted focus on promoting promoting biomedical research through primarily through supporting primarily undergraduate institutions, and they have mandatory focus on training and teaching and providing support for data science and bioinformatics. And this is what I call socializing learning for data science, because we'll reach more than 50,000 undergraduates. And what we're trying to do through NEPI and through these programs is really provide free or very low cost training in bioinformatics and data science. So we also have the IDEA networks for clinical translational research, the IDEA CTRs at 20 active states that focus on translational research. We also support, the NIH supports co-funding for R1s and R15s and STEM applications that come within region of being funded by Categorical Institute. And we do have 4 hubs that promote entrepreneurship within the IDEA network. So the IDEA networks themselves, and I will talk about some of the specific things that NIH is doing to promote data science and computational biology through cloud computing training and through Google and Amazon contracts. And the CTRs are clinical translational centers that involve multiple medical schools within the IDEA states. And there's a co-funding coordinator that has been used to really help people within states to achieve funding through a categorical institute. And of course, the IDEA Regional Technology Centers have seed programs, and they teach business fundamentals for technology core directors for the IMBRE and COBE programs. There are a number of other collaborations and participations in NIH-wide initiatives. I'll go briefly through this, and you'll have the slides to see this. These are really from a recent town meeting that we had with NIH. Dr. Ming Li presented some of this information. So there is collaborations among the IDEA programs. There are supplements that are being given to COBRE CTRs for a number of different purposes, including a number of them for COVID-19 research. There are collaborations with non-IDEA NIH programs, including the maternal and infant mortality research, the COVID-19 patient registry, cloud computing for biomedical research, and the NIH STRIDES program that involves access to Google and Amazon cloud computing, and I'll say something about that a little later. We also have competing participation in competition for NIH-wide research initiatives, including the active files for COVID-19 therapeutics, and the rapid acceleration of diagnostics for undeserved populations. So a new program for NIH that I think will be important for some of you to know is the SURE program, which is Support for Research Excellence SURE program. And this is really to target primarily undergraduate institutions that serve minorities. And it has different programs. One is to support investigators for NIH research funding and to support research excellence as a first independent research award, and to build administrative infrastructure, broadening participation. And there are specific qualifications for this, up to 25% of the students being served by Pell Grants. It replaces a number of the opportunities that were provided initially for minority institutions. So a number of really small, primarily undergraduate institutions can compete for these awards. Of course, the INBRI programs within state will assist in any of those primarily undergraduate institutions within these 23 states in Puerto Rico to submit these competitive awards to NIH. This, again, the map of the IDEA program states that, as I told you, involves the COBRIs, INBRIs, IDEA CTRs, 23 states in Puerto Rico, labeled here in this map as green. Really, they are the states that have a great population, a lot of interest, unfortunately not the resources some of the bigger states have and the elite programs throughout the country. And NEPI is facilitating accessing this information and representing all these IDEA states to NIH and to the federal government. So NEPI has done a number of initiatives, and we are out in the process of developing more. Let's see, get this out of here. We're providing funding for IDEA undergraduate students to travel to Washington, D.C. Every other year, we have a national meeting with over 1,000 people attending in Washington, D.C. And we'll provide travel funds for that. We're awarding awards for undergraduate, graduate, and assistant professors for best presentations in our regional meetings, which are also every other year, overlapping every other year regional meetings, and then the national meeting. The access to IDEA-wide databases, and we provide free access to virtual training courses and exercises created by IDEA faculty, and discounts to other virtual courses in data science and informatics. We have really have a collaboration with Pine Biotech, and we're providing a number of these courses free of charge or with low charges throughout our network, accessing more than 50,000 students. We also going to have job postings and job searches in the IDEA program. We also were proud that we are co-sponsors for this particular symposium, which provides, I think, tremendous information for what has been happening in the field of data science and training. We're also very proud to announce for the first time that we are the recipients of the Tober Foundation support, a donation that will help us gather data science, bioinformatics content, including training modules and lectures, and we'll disseminate them through our website to all the NEPI stakeholders and participants. And we're going to provide Pine Biotech material for free or through discounted prices to all NEPI members. We'll also be providing CAS awards, as I was telling you, for different conferences that we support. And we also will be covering expenses with this donation for a general lecture on citizen science, which is also very important. And we'll be covering expenses for launching surveys and maintaining our NEPI website, and engaging the NEPI members and organizations into a larger discussion about how best to train people, especially in data science and bioinformatics, with a goal to increase competitiveness for biomedical research. I want to very briefly to also mention our own program, the Louisiana Biomedical Research Network. This is the website here. It does have a number of important information and recorded lectures on coronavirology and other symposia that we've held together with Pine Biotech. And we've also been involved heavily in summer training. And this is the previous summer program that we launched with over 10 different types of programs, a number of them virtual with the assistance of Pine Biotech. And you'll find this information in the LBRN website. And we were very proud that we had a number of participants doing specific projects as well as virtual projects through this. I want to take a really brief time because I haven't seen that mentioned here, that obviously as you'll gather through my presentation, that NIH is very much interested in really disseminating information and training people, the new generation of scientists in biomedical research, especially in data science and bioinformatics. And there's a special office, the NIH Office of Data Science Strategy, that has been created. This is the website here, datascience.nih.gov. I think very important for any of you that are interested to really look at that site, because it has a lot of very important information, as well as free webinars and so on. And of course, this is, as everybody knows, the explosion of NCBI data and services is noted here. So the amount of information we are handling is really humongous. It has been noted already. And that really necessitates the need for training people to be able to really understand these really large data sets and be able to derive information that will help for biomedical research. So the strategic plan for data science, which is really on that website that you could download it, it notes that our nation and the world stand at a unique moment of opportunity in biomedical research, and data science is an integral contributor. So NIH recognizes how important this is, and that's why it creates these programs and trying to engage the entire country, not necessarily only the top elite institutions. So this is a definition of data science in the disciplinary field of inquiry, in which quantitative analytical approaches, processes, and systems are developed and used to extract knowledge and insights from increasingly large and more complex data sets. And this is part of the third principles, which is really to create data sets that are findable, accessible, interoperable, and reusable. And you can see all these qualifiers within the website for the strategic plan of NIH. A very important document that really gives you a blueprint of what NIH will be focusing on the next 5 to 10 years and beyond. So this is the strategic plan for data science. And it looks into these components for data infrastructure, to modernizing data ecosystem, to data management, analytics and tools, to workforce development, and stewardship and sustainability. And the IDEA program is particularly focusing, and NEPI's efforts are focusing on workforce development. We need to enhance the NIH data science workforce, expand the national research workforce, and engage a broader community. Because I don't think if we really do not socialize these tools and information and training and education, we won't be able to compete at the international level with other states and countries that are really focusing a lot on training their people and competing at large with the United States. So Science in the Cloud is a very important program. The NIH created the NIH Data Commons. It's as a pilot program, and that really creates access to test data sets, including the National Heart, Lung, and Blood Institute's Transomics for Precision Medicine, TopMed, and the NIH Commons Fund Genotype Tissue Expression. So these data bases are really available for mining by all people. And this is part of really socializing all these data sets to everybody who may be interested in them. And one of the more important things that NIH is part of the strategic plan is citizen science. And really citizen science aims to bring science and science policy processes to the public, to ensure that science is responsive to public concerns and needs, and to harness additional resources to capture data and innovation in a wide variety of fields. And a couple examples of specific modules have been created. The iWire, which is an online resource launched in 2012, that challenges players to map the three-dimensional structure of neurons. And you can find more information about that at the NIH website. And the experiential citizen science training for the next generation program that promotes biomedical scientific careers among K-1 through 12 students, and aims to increase participation of individuals and groups underrepresented in biomedical sciences. So it was very wonderful to hear CMU's elite program, Georgetown. Our goal is to be able to really spread that information and engage the entire country. As specifically to the cloud computing resources now being available for IDEA states, 23 states in Puerto Rico, and minority serving institutions. And this is the NIH STRIDES program, science and technology research infrastructure for discovery, experimentation, and sustainability. And this is an agreement of NIH with Amazon, Google, and Microsoft to be able to provide discounted cloud services and training through these agreements. And there is now a working on understanding what the needs are. And there's an RFI that will expire in September to find out input for current and future needs and utilization of cloud computing from institutions within IDEA eligible states, RCMIs and MSIs. Part of the idea is to provide resources to the cloud to a lot of these institutions that do not have local resources for computational biology. And there are NIH workshops on broadening cloud computing. The next one is September 13, 14, and there is a website for registration for all IDEA participants. And there are 30 free cloud training days, 15 by Amazon, 15 by Google. These are the specifics on the number of students that could participate. And there's a direct contact to NIH for those people that are within those 23 states of Puerto Rico that they could participate. So I want to leave you with this. This is my last two final slides. I would like to quote of people this quote by Harriet Beecher Stowe, that common sense is the act of seeing things as they are and doing things as they ought to be done. We are striving very hard to socialize training in data science and bioinformatics, and hopefully we'll lift all our communities to be able to be much more competitive for biomedical research. And this is a picture of the LSU stadium football few years back when it was actually full of people. We hope to return back to normalcy soon, and I would welcome any of you, if you would like to visit LSU, to also visit us at the BRN. Thank you.
Thank you, Dr. Casoulas. We do have a couple of questions. So the first question is about members of LBRN about applying to the IDEA co-funding for R15. What is the process for that? Yeah, so typically, obviously, we assist in this through our administrative core, but there is a specific contact person at NIGMS, and they need to know. And when you are really within potentially close to a categorical funding, they're nominated to be cross-funded by NIGMS and the Categorical Institute. And typically what happens is NIGMS and the IDEA program picks up the first year for an R15 or the first 2 years for an R01, and then the Categorical Institute picks up the remaining funding. Part of the idea is to allow the Categorical Institutes to fund proposals that did not make the cut through the Categorical Institute. So typically, we've seen grants within 5 to maybe 10 percentile points, depending on the topic and the interest by NIH, that could get funded for these 23 states in Puerto Rico. Okay, thank you. And another question is, do you collaborate with international institutions for research, faculty development, student exchange, etc.? Yes. Well, you know, the home unit that really administers the Louisiana Biomedical Research Network is the Division of Biotechnology and Molecular Medicine at LSU, a School of Veterinary Medicine that I direct. And we are working to export a lot of our training in collaboration with Pine Biotech. We have talked to a number of countries we're interested in Latin America, as well as Europe, Southern Europe. And quite soon, we'll be announcing a series of training modules that are provided right now to only LSU students internally, but we hope to export these training modules, exercises, potentially a virtual master's degree to international students
with a focus in Latin America and Southern Europe, but this time, potentially Middle East and elsewhere.
All right, thank you very much. And final question is, what do you think is kind of a way for students without any bioinformatics background, so from maybe a life science background, are there any summer opportunities for them to join some of this training?
So within these 24 states, it's fairly easy because all these INBRI programs, the Idea Network of Biomedical Excellence, accept undergraduate students that within those states. In Louisiana, we have 27 institutions that participate in the summer program, and they apply and they get in and they do a summer research, and not only in bioinformatics, but also biomedical sciences by really being hosted. A lot of things the last couple of years have been done virtually, very successfully. If you're not in an IDEA network, the only opportunities would be for us, at least to LSU, our current efforts to export this in a larger format. And NEPI is interested primarily, obviously, for the 23 states and Puerto Rico, because this is really our charter to help those 24 states. But ultimately, we believe that we could sort of socialize a lot of this training because there's a firm belief within the network that these tools and training should be available to people at very low cost and as many people as possible, as young as possible, because that's the only way to really have an inversion point, as people say in business, to really be able to really transform education. You have to reach out to as many people as possible and as young as possible.
All right, so final question, thank you for that answer. Are there any opportunities for people working with plant sciences, agriculture, and plant breeding for learning about bioinformatics and potentially joining research projects?
Yeah, obviously at LSU, we have the LSU Ag Center, a number of the students participate in plant-related bioinformatics, computational biology projects. The students enroll at LSU and the College of Agriculture, and we have they participate in a number of our projects. I think it's very possible that we create certain virtual projects that relate to plant science specifically. And we have a number of researchers that are working on advanced bioinformatics tools and data science for plant genetics and plant-related projects. So, data science and biology includes both plant and eukaryotes in so many ways. So the same tools are used across the board, whether it's a plant or it's actually another organism.
All right, great. Thank you very much for this wonderful presentation. So Sonalika, I'll pass it on to you to introduce the next speaker.
Thank you so much, Ilya. And thank you so much, Dr. Gus, for telling about the various opportunities that are there at the LSU. Now, I would like to welcome Dr. Shindo Hicks, who is a program director for Bioinformatics Research Program with a focus on health and health disparities. Let me just share my screen. Yes. His research interests include bioinformatics and computational genomics, multi-platform and multi-scale data analysis and integration, genomics and epigenomics of common health diseases, pathway prediction and modeling gene regulatory networks, as well knowledge and drug discovery using big data. And Dr. Shindo Hicks would be giving a talk on bioinformatics and computational genomics research and training. Over to you, Dr. Shindo.
All right, yeah, sure. Thank you very much for that great introduction. And I wish to thank the Pine Bio organizing committee for allowing us to share the information about the activities going on in our lab. So I guess I'll wait for Dr. Kasuras to exit before I share the screen.
Yes, Dr. Shindo, you can share your screen. You have the permissions.
Okay. I guess the screen is not being shared yet.
It was shared and then I think it went away. Maybe you can try.
Okay. So let's see. So can you see my screen?
No, we can see your video, but we don't see your screen.
Okay, well, what about now?
Now we can, yep.
Okay, let me see. All right, is this okay now?
Yeah, maybe you need to share the other screen. We see your view, not the presenter view.
Okay. That's weird. So what about this?
Yep, perfect.
Okay. All right, so thank you very much to everyone, and thank you very much for inviting me to share the information about some of the activities that are going on the at the research center here in New Orleans. So I'm going to focus mainly on the bioinformatics and computational genomics research and training program opportunities that are here in Louisiana. I would like to mention also that we are part of LSU, just like Dr. Kasulas talked about. So we are basically here at the medical center that we are in. And then a lot of the information that I'll provide, obviously, if you have questions, you are also welcome to reach us through, you can either call me or by cell phone or by local phone or by email, any medium that you present. And in the event that you miss some of the information, you can also just Google LSU Bioinformatics and you will find us there where we have various programs that I'll be talking about. So what I would like to talk about mainly is the bioinformatics and genomics program, and I'm going to focus on the opportunities under the three missions of the university. The first mission is the research mission in the basic department with Department of Genetics. So research for us is a core mission. We also have research under the service support, which essentially means that apart from conducting our own research, we also provide other investigators in the state and also in the region with bioinformatics service support. The other part that I'll talk about is the education mission, where we have various opportunities. We have a newly established master's program in bioinformatics, which is a degree program, which essentially means that you can get informal training, say, for example, through Pine Bio, and then you can also punctuate that with a formal training program where you actually get the degree. So essentially, for example, Pine Bio would provide you with a foundation, and then you can come here and undergo formal training and get the degree. Or we'll basically discuss with Pine Bio to see maybe how we can manage those programs. There's also a summer program in bioinformatics, which is a funded program. Essentially, you can just apply to that program, and then we will select you if you qualify. And basically, you work with us for 10 weeks. And during those 10 weeks, you get a paycheck. Therefore, you don't have to go looking for a summer job for that. We have the outreach program, which essentially means that we train students from neighboring institutions, especially historical Black colleges, like Xavier, Dillard, Southern University, and others. We also have the international program, which we participate in with the various countries, particularly most at this point, mostly with the African countries. And then we are involved in technology development, which I'll also talk about a little bit. So this is a program. We are a very new program and very few people, but we leverage our expertise here with expertise in at the neighboring universities or with other collaborating partners internationally and in the region. So there's myself, the director, there's Dr. Wu, senior bioinformatician, and there's Aditi Pucci, who's a bioinformatics analyst. And we take bioinformatics, as I said, bioinformatics and computational genomics graduate students. So right now we have David Odoknoy from Nigeria, who recently joined our lab. We have Tarun from India, and a joint program with UAB. The program itself, as I said, is located at the medical center here, and this is our website. And through this website, you can click on each one of these pull-down screens, and you can find additional information, including our publications and all the activities that we are involved in. We do, as I said, we provide bioinformatics services. We conduct genomic research, a lot of collaborative research, and then education. And you can reach us through here. So in terms of setup, this is basically the way our lab is set up. Obviously, when you sit, you basically sit like back to back. You don't face like one direction, you sit back to back. But our lab is set up this way. The reason I'm showing this slide means, like Dr. Kasulas mentioned in the previous presentation, we do have the capability to collaborate with partners around the world. So we get data from around the world through various platforms. For example, right now we collaborate with the University of Cape Town in South Africa. We can get data from there. We do use the Globus platform that allows us to perform collaborative research and collaborative data sharing. The Globus platform essentially means that we can move data from anywhere on the globe to our lab using the Globus nodes, provided that the other end has also got the Globus node. So if you are working with us, this would be the setup, whether you are a PhD student or whether you are in our master's program, or even if you are in the outreach program. So in terms of research, basically, this is basically the 5 domains of research that we are involved in, but I want also to mention that we are not restricted to human research. We can do any type of bioinformatics, whether it's the animal sciences, plant sciences, we can do. It's all genetics. And fortunately, I have worked in both the humans as well as the animals and plants. So therefore, I feel I have an understanding of what is the bioinformatics. Genetics is genetics, whether you are working on animals or plants or humans. It's just a matter of semantics of how you can formulate the question that you would like to address. But these are the programs that we work on. In addition to the listed, the 5 programs that I show here, you'll see a green line showing here simply means that this is predictive modeling basically, where we are using machine learning and other algorithms to predict different diseases. For example, we are working quite a lot with the Alabama Genomics, Initiative, where we genotype a lot of people, and we are using genotype information to basically develop polygenic risk scores. Essentially means that can we predict the disease risk of an individual, or how to stratify those patients. And that also involved that although these are divided the quadrants here, we do a lot of data integration in my lab, that we integrate various pieces of information so that we can improve the outcome or improve the predictability of the algorithms by basically refining them. An example would be, can you integrate genotype data, for example, with the RNA-seq to refine the predictiveness of that algorithm. So we also develop technologies, of course, as I said, we support other people other than our own initiative for research. So we have developed the pipelines. Basically, these are the pipelines that those who are collaborating with us and those who need access to that, to us through service support, can access this. So the pipeline that I've listed here, basically, they are not all of them, as you know. Every bioinformatics question depends on the investigator, but these are the main ones. So we have capabilities for microarray data analysis, and also genotype data analysis, essentially for microarray platforms. We have RNA-seq data analysis pipelines, exome sequencing analysis pipeline, for genome sequencing. We have recently developed a single-cell analysis pipelines, and we have developed also methylation analysis pipelines. These pipelines that I'm listing here are backed up by our publications so that the investigators who are collaborating with us can actually see that we are not only saying that we can do this, but we actually have expertise in this, and those are demonstrated expertise, which is very critical, as Dr. Kasulas mentioned, that is very critical for grant applications, because you cannot just list the names, but they want to see, can you actually do that? Or is there any evidence that you have actually done that? As you see, these are disaggregated algorithms here, or disaggregated platforms. They are nowhere near where PineBio is. PineBio has an integrated environment. We haven't reached the integrated environment, obviously. So, but this serves us well partly because we are also researchers. We are not only like developers of the tools. Therefore, our algorithms, for example, can be leveraged with, say, the PineBio, the TBio platform. So, what do we do? So these are other expertise that we have. We do a lot of copy number variation, mutation profile, mutation spectrum, and the mutation rates, methylation profiling, RNA sequencing, pathway analysis. So these are some of the things that we can do. Obviously, we can also do some other, which are outside this outstream. For example, we could do ChIP-seq data analysis. We do a lot of proteomics data analysis here. Because we have actually an active proteomics core here. So I just wanted to give you a little bit about some small demos about the things that we have done. As I said, we have done a lot of genotyping analysis. So here's an example of what we can do. So for example, we have this is genotype data from 350,000 cases and controls. Basically, these are genotyped people. Now, we don't have to genotype these ourselves. Basically, we can get data from public domain. For example, this data is coming from dbGaP, which is basically at the NIH, everybody has access. So from there, we can analyze here, as you see on the right side here, we can analyze basically the SNPs or mutation, germline mutations. And what you see at the top, these are simple chromosomes. This is just a Manhattan probe. Plot mapping the SNPs to the chromosomes, and on the right side here you can see the log p-value. Essentially, we can use this is the threshold here. So basically, we can compare cases to controls and identify those mutations. Fortunately, this was what we used to do like 10, 15 years ago. But now you can do sequence-based genome-wide association studies. You don't have to do genotyping. So for example, here's a cartoon showing exactly what I just showed you before there. This is just a cartoon with basically different SNPs mapped to different chromosomes. And the color code simply means for different diseases. For example, it would be cancer, heart disease, or anything that you work on. So this is simply aggregating the SNPs, mapping them to the chromosomes, and then we can do this. So this means that you can also do analysis across diseases or across cancer types or anything that you are interested in. Or you can use these resources, which are also available at the NIH website, that you can also use these resources as a reference resources. But I wanted to highlight these are the SNPs that we are actually using for developing algorithms like polygenic risk scores. As I mentioned, we also whole genome sequencing here. As you can see, we work very much on triple-negative breast cancer, and here basically what we are looking is sequencing different genomes of breast cancer and comparing it to different types of non-triple negative cancer. Triple negative cancer, for those who don't work in this area, simply means that it's negative for the three markers: estrogen receptor, progesterone receptor, and the human epidermal receptor growth factor receptor. So if it's negative for that, then it's classified as triple negative cancers. But the basic premise here is sequencing the genomes of people who are diagnosed with triple negative cancer and comparing to determine are there any differences between the genome, the triple negative genome, and the other genomes. So basically, you can work with us on that. And then you can see here basically classification of different mutations, different genetic alterations, and basically the range of base pairs and things like that. So these are things that you can do with us in terms of collaborative research. Again, it doesn't have to be that you work on triple negative cancer, you could be working on something entirely different, but we'll be glad to work with you. This is just the functional based classification of mutations. On the left side here, you can see the genes here and different mutations. For example, in this case, we have copy number variants, point mutations, basically like SNPs, and then simply mapping them. If, for example, you have exome sequence data, you can map them to the 3D structure and determine where they're located. Are they inside, on the surface of the structure, inside the structure, in the backbone, or whichever way you are studying. That becomes very important for the people who are studying drug development. So you can also do functional analysis to look at where they're located. Do they actually disrupt the enhancers or promoters? And are they located in microRNAs? So we have actually looked at this problem in terms of looking at SNPs, whether they disrupt the alternative splicing. In that way, we have actually published in our previous reports. We do a lot of data integration on a small scale, definitely nowhere again near T-Bio, but we do integration of germline and somatic mutations. So here we have the germline genome that I just showed, and then we have the cancer genome, and then basically we integrate that information. And then we can look at, for example, do genes which contain germline mutations also contain somatic mutations? And for example, do pathways that control the germline genome also controlling the cancer genome? This is going to be very important as we move through predictive modeling, where we have to look at the genetic predisposition versus the disease itself. So that's other than just predicting the outcomes, which we do now with the, say, tumor data, we may also be to start looking at how to predict the disease risk. And that means that other than focusing on what is now called precision medicine or genomic medicine, we may also have to be looking at the precision prevention. For example, if we can predict genetic predisposition that is linked to a disease. So that's how we are looking at that. We have also introduced a new dimension to this. Other than looking at the germline and somatic mutation, we have also introduced the epigenetic data there. So for example, in this cartoon, you can see here, we have gene expression data here, we have methylation data, and also the genotype and the gene. The rationale here is basically to determine where the somatic mutations or germline mutations are located are those methylated regions. So that is very important to determine whether methylation actually does mediate the interactions between the germline genome and the somatic genome. So also, as you know, epigenetics is modifiable. Therefore, if we could determine that, therefore, things like studying like the biology of behavior, that might become quite interesting in the sense that there are certain factors that could be modifiable factors, for example, obesity and other morbidities that can be modified, and how that relates to epigenetic regulation, because we can't modify the germline genome, but we can modify the other factors. So that's the research part. So what about the service part? The service part, basically, we have various ways. We have the various models. One is through collaborative research model, where we write a grant together with you, and then basically we are all on the grant. The other part would be where we basically just do a project-based analysis where we say, well, this project, maybe the investigator says, I don't have enough money, so I can't budget for bioinformatics. I can budget for bioinformatics, but only a small piece. Then we'll say, okay, so this is a project whereby, so for example, this could be pilot studies. A lot of postdocs and students do pilot studies. They are funded by LBRN or other organizations. And maybe we'll say, okay, just budget, say, $1,000 for bioinformatics. And that will be project-based collaboration. Then there's a fee for service where we'll say, okay, I'll just pay you a fee. You do the work for me, tell me how many hours it will take, and I'll do fee for service. So in this cartoon, I'm just showing that everybody who does service support knows that this discussion always happens whereby the bioinformatician goes there with their laptop and the physician on the other side here and the other guy. So there's a stalemate there. The bioinformatician wants to know what is the objective of the program, or what is the outcome of the analysis. And on the other hand, the physician is saying, tell me, how many samples do I need? Or those kind of things. So those discussions are there. So on the far left here, I'm showing that there's a table here that we have. So that is something that we want to collaborate, resolve the program before actually the project takes place. So that's the collaboration. We do have other collaboration here, which is the collaboration with the precision medicine program, which is also here, where we have the information coming from the physicians ordering the tests, and we are doing the bioinformatics variant calling and giving them the report card there, and then they can report that back to the patient. So that's the service part. Then we also have been working on using the service part. Also, we do data mining. For example, in this cartoon here, we are helping the people to use the COVID genome atlas as a validation to the experiments that they themselves performed. So recently, we just published actually a COVID paper on that from Louisiana, using this COVID genome atlas from Harvard as the validation procedure. So the bioinformatics opportunities here, we do have, as I said, the master's program here is a joint program between the graduate school and the School of Medicine. It's a 2-year program with 10 courses plus a thesis. It's a basically project-driven program. Basically, it has 36 credit hours. And these are the faculty here. So we have the co-faculty basically who teach that. It's a basically interdisciplinary program. I teach bioinformatics and computational genomics. Dr. Wu teaches the bioinformatics programming. Dr. Taylor metagenomics. Dr. Lin biostatistics. Dr. Hollenberg and Dr. Mando, they teach molecular genetics, again, biostatistics here. And also Dr. Gide teaches things like things where compliance. Then we also, in addition to that, we have the program is enriched by bringing the external, like Dr. Phillips said, or Dr. Campio said. So the program is really enriched by bringing people with expertise from outside, so that the students don't only listen to us, but they can also listen to other people who are very well accomplished from outside. For example, here, Dr. Jonas Almeida is the director of the Data Science Service Center for the MCI. Mohammed Tayoun is in industry, and Dr. Jay Pak from actually LSU Center for Computational Biology, and many others that we bring them here. So this is basically to enrich the program, and so that students can also discuss what are the other opportunities. For example, under this same program, we could bring in someone like Dr. Campio and say, okay, can you, for example, enrich our program by telling our students what are the opportunities outside there, other than what we tell them. These are some of the students, the graduate students who have graduated. Yes?
Sorry, we're just a little bit over time. And before we have a chance to, do you have a lot more slides? We have some questions that people have.
No, actually, I'm almost there.
Okay.
I think I've probably one slide or two there. So here are some of the graduate students who have used our program, who have graduated from our program using bioinformatics services, and some of the new students here. And this is the summer program here, where basically all the students that we have trained under the summer program. So I don't need to spend a lot of time on this because this information actually is on our website. But I wanted to share with you that actually we do these things, and we list where these students are. They have graduated from this program and where they are now. This is an international program between us and South Africa, Zambia, and Tanzania recently. So we are involved in different programs around the globe as well. I do participate myself in the BioNet, which is part of the H3 Africa training program. And these basically are the funding agencies. And with that, I would say thank you. And certainly, if there are any questions I can answer, or maybe I can answer at the end of the program. Sorry for being too quick on this one, but again, this information is available. You are welcome to reach me, even through Dr. Kasula, so through Ilya, you are welcome to reach me if in case you lose my information. Thank you very much.
Okay, thank you very much, Dr. Chindo Hicks from Louisiana State University Health Sciences Center. So we do have some questions. I think you mentioned some of the answers already here, and we will connect you to some of the people that have those questions, primarily about international collaborations. So we will direct them to you. And Sonalika, please, if you can introduce our next speaker.
Thank you so much, Ilya. Now I would like to welcome Dr. Chris Taylor. He would be talking on the bioinformatics research and education for medical and graduate students. Dr. Chris Taylor's research lies in the realm of computational biology and bioinformatics, specifically related to applications of high throughput sequencing. This research is highly collaborative in nature, involving the development of algorithms for analysis and visualization of sequencing data. Over to you, Dr. Chris.
All right. Let me get my screen share up. So can you guys see my screen?
Yes, Dr. Chris, we can see your screen.
All right, great. So yes, as mentioned, I am an associate professor in the Department of Microbiology, Immunology, and Parasitology at Louisiana State Health Sciences in New Orleans. It's the same institution that Chindo, who you just heard speak, is from. I also serve as the director for the Louisiana Biomedical Research Network's Bioinformatics, Biostatistics, and Computational Biology Core. And that network is headed up by Gus Kasoulas, who spoke just before Dr. Chindo Hicks. So thanks to the organizers and to Ilya for inviting me to speak. And I'm going to just discuss some of the bioinformatics research and education opportunities that we have for medical and graduate students, and how we sort of address the issues of teaching bioinformatics in a medical institution. All right, so my background is in computer science. So I got my undergraduate, my master's, and my PhD in computer science. And during the course of studying my PhD, I transitioned into computational biology. And this was a time when the human genome was just being finished, being sequenced. And one of the things that was occurring was that there were a lot of specialists who were studying bioinformatics and computational biology, and they came from all different backgrounds. So some people were from mathematics, some from computer science, others from biology who got into computation. And in that time, it was really, bioinformatics was very much of a niche landscape where there were a handful of these specialized researchers. But nowadays, nearly every discipline within biomedical research has the needs for bioinformatics methods. And we've really narrowing this chasm between the biology and the computing sciences. And I try to stand sort of in between the two so that I can interact with people on both sides of these fields. So the challenges that I have run into is that as high throughput sequencing has come onto the scene, starting in 2005 through about 2008 getting established, and then in the last decade has really exploded in the amount of different things that you can use it for, because the cost of sequencing come down so much. There's a plethora of sequencing data that needs to be analyzed, and it can't really be served anymore by these niche researchers in bioinformatics and computational biology. So I've found that there's a need for training biomedical researchers, particularly, to empower them to be able to perform computational data analysis on their own data. And those researchers really don't need to be bioinformatics specialists anymore. They can actually be biomedical researchers that learn enough to do analysis of their own data. The problem that arises is really that these researchers come from a diverse set of backgrounds. Some of them have had no computer programming experience whatsoever, whereas others have been programming as a hobby, for quite a bit of time. And so the traditional format or educational programs where you take people through step by step can end up being, in a course setting, too fast for some of the people who haven't had this, experience, but can also be incredibly slow for people who have had that experience. And so when you get one of these courses and you have these students with all these varying backgrounds, it's very challenging to do a traditional lecture course. So the emerging solution that we've come on to and have partnered with Pine Biotech and the Louisiana Biomedical Research Network is more of a reliance on technology to provide online learning opportunities, which will allow students to move individually at their own pace. If they can move really quickly through the modules, then they do them fast. And if they need to take some more time, they can take a bit more time. And this is really something that you can't really accomplish in a traditional in-person lecture format, but with online course modules, it allows people to move at their own pace. And so about 4 or 5 years ago, I developed these lecture series that I conducted, and I conducted them in person at LSU Health Sciences New Orleans, but we recorded all of the lectures using this media site capture technology. And we broadcast them also live streaming for remote viewing for people at LSU and other places throughout the LBRN network. And then there was participation via group forums for questions and announcements and distribution of course material. And then we did some quizzes and tests to test the understanding of the material. And the idea behind this was always that these recorded lecture series would persist and be available for viewing on demand. And so the 2 lecture series that I gave in 2016 and 2017 are still available on my metagenomics server at one of them was a general introduction to informatics, and the other was an introduction to microbial community analysis. And these are all available as slide sets, videos. All the other lecture material is available in archive for download at those above URLs. And interestingly, some unintended consequences occurred, and I got an email out of the blue from the Northern New England Clinical and Translational Research Network. And I guess they had done some searching and they had happened upon my lecture series. And so they asked me for permission to link the lecture series for their own investigators instead of, reinventing the wheel. And so I think this is, one of the very powerful aspects of the online learning paradigm is that you can reuse these materials and don't have to continually reinvent the wheel and give lecture after lecture on the same course material. And when we held these lecture series, we also, throughout the LBRN network, provided them to our primarily undergraduate institutes for usage. And they could take the lectures that were recorded online, and then they could add their own sort of lanyard to them. And so this was done at both LSU Shreveport in northern Louisiana and also at Southern University in Baton Rouge. And they enrolled students in courses at their own university. And a lot of them were just topical courses. And then they allowed the students to use the remote lecture material, but then the instructor, who was the instructor of record at each individual institution, would partner and would provide in-person lectures to supplement what the students were learning based on their needs. And so that was done at those two institutions. And then since then, the LBRN has partnered with Pine Biotech, and Pine Biotech offers a whole library of courses that are available online through their EDU platform. And those are courses that students, again, enroll and they take at their own pace, but then they're also, if you're enrolled in the LBRN programs, are supplemented with some in-person meetings that are done online as well. So this addresses part of the critical need for education in the field of bioinformatics. And so this Pine Biotech program, it was initially established in the summer of 2018 in that partnership with Pine Biotech and the Tauber Bioinformatics Institute. We had obtained 25 educational licenses as part of the LBRN, and we provided them to each of our primarily undergraduate institution campuses, and they were able to enroll in the Pine Biotech educational modules and take them. And so that pilot program went so well that it's been expanded into a full-blown partnership with Pine Biotech. And the students that take these modules on genomics, metagenomics, transcriptomics, and other topics, they are encouraged to present their project posters at the LBRN annual meeting and other meetings that are held, a lot of them virtually now due to the COVID restrictions. So that's been a large partnership that has allowed the kind of expansion of the ability to teach bioinformatics to individuals who have very diverse backgrounds, and they can go at their own pace through these course modules. So this has sort of been a whole evolution over the past 5 or 6 years. And here's just a snippet of some of the current offerings from those Pine Biotech modules on transcriptomics, genomics, metagenomics, and epigenomics. And this is all apropos to the Omics forum that we're going through right now. So one of the other things that I'll draw your attention to is that as part of the LBRN and the bioinformatics, biostatistics, and computational biology core that I direct, we hold an annual conference on bioinformatics and computational biology every year. And we just held our 8th annual conference. The 8th annual conference was originally scheduled for April of 2020, but due to the emerging COVID-19 pandemic, we didn't have time to pivot, and we actually had to cancel it. So we held our 8th annual conference virtually April 15th to 17th of this year. And we have the recordings from all of the past talks are available online through the LBRN portal on YouTube. And the links are available at the LBRN conference on biology and bioinformatics link above. And part of the meeting that we held just this year was a session that was entirely organized. It was a whole day organized by Todd Riley at the NIH Center for Information Technology. And this was discussing the NIH's STRIDES initiative, which is a cloud computing initiative whereby they've partnered with Amazon Web Services Research and Google Cloud Research. And they provide training, they provide cloud credits for researchers at Embry institutions. And we've been participating in a lot of these trainings over the course of the last couple of months since they've been established. And then we also had other eminent bioinformatics and computational biology speakers who covered topics across the gamut of computational biology, including pandemic modeling of COVID-19, the gut microbiome, herpes simplex virus, and the role of statistics in epigenetics research. So this is just a snapshot of all of our primary speakers that we held at the 2021 conference. And the 2022 conference will be held in April of 2022. So keep an eye out for that if you're interested. We're not sure quite yet whether it will be a virtual format, whether it will be back to an in-person format or some form of hybrid format. So we'll see how that goes. So zooming in a little bit, that's a lot of the initiatives that I'm involved with on a broader scale. My own research lab at LSU Health Sciences, when I built it back in 2012, I built it with this open lab layout in order to promote collaboration. So we don't have, cubicles or partitions or anything. And I mean, that was a great idea back in 2012. Maybe it's not such a good idea nowadays. But the idea was that, if somebody's sitting there working on a problem and they have an issue, they could turn to the person next to them and see if they had some advice. And so I always have a mix of different researchers in the lab at different career stages. I've had postdocs, I've had graduate students, I've had undergraduates through our summer research program. I've even had high school students through our summer research program. And even a few junior faculty members who wanted to come in and learn how to analyze their own biomedical research data. And so, we work with them, and it's sort of this, the old proverb of if you give a man a fish, he can eat for a day, but if you teach a man to fish, he can eat for a lifetime. So the idea is we teach these researchers how to analyze their data, and then they can kind of spread their wings and do it on their own in the future. And so I primarily, in my own research, I work primarily with MD, PhD students who go through both our medical program and our PhD program, and it's a joint program. And we just graduated Vince Maffey. He was an F30 fellow working in alcohol research, looking at the gut microbiota. And I currently have in my lab Jacob Elnager, who's just submitted an F30 grant, which we hope will get funded. And he's working on shotgun metagenomics analysis of the vaginal microbiome with specific emphasis on bacterial vaginosis. And then I've just hosted for a rotation a student, John Lammons, who did a rotation this summer and likely will join my lab as another MD PhD student. We've had several past members of the lab. As I said, many undergraduates, several PhD students who work with other investigators have come in, and I've helped to co-mentor them if they have a project in their home lab that involves DNA sequencing analysis. And so in terms of ongoing research projects, my research really focuses in heavily now on the vaginal microbiota and various sexually transmitted infections. So I have a multi-PI R01 grant that is studying the clearance of chlamydia prior to treatment. So we have some women who come in, they get tested for chlamydia, they end up positive. By the time we call them back in for treatment, they've actually cleared the chlamydial infection through natural host defense. And we're looking at the properties of the microbiome that potentially promote that natural clearance prior to antibiotic treatment. We've also got a new R01 that's based in South Africa and is joint with the University of Cape Town and the University of Southern California. And that's looking at adverse birth and newborn outcomes and the interaction of the vaginal microbiome with preterm birth and adverse birth outcomes. Then there's an R01 that I have with a collaborator at UAB, Christina Musney, to study bacterial vaginosis. And my student Jacob, the F30 that he submitted, is really working jointly under that grant. I'm also a member of the Center for Clinical and Translational Research at UAB. LSU Health Sciences is one of the sub-sites for that. So that's this UL1 grant. And then we're doing some work on the gut microbiota and precision medicine under a UH2 grant. And we just got an R21 got a fundable score last month. That this is going to be kind of an interesting one. We're going to look at transgender men who are just initiating testosterone use And we're going to look at how that impacts the vaginal and reproductive tract microbiota and the propensity for getting bacterial vaginosis after testosterone use. And then, as I mentioned, I'm also part of the Louisiana Biomedical Research Network. And more recently, we've gotten an NSF grant funded for a high performance computing cluster. So we're just into the start of the second year for that. And that grant has supported the acquisition of a large computing cluster, which we call Tigerfish. And it's it's actually the first high performance computing that has been broadly available for our Health Sciences Center campus in New Orleans. We've had access to some of the clusters in Baton Rouge and through the Loney network, but this is for New Orleans specifically, and it provides up to 193 teraflops of compute, 1,400 total compute cores. It's primarily composed of these compute nodes, each of which has 192 gigabytes of RAM, but it also has available a large memory node, which has up to 1.5 terabytes of RAM available for tasks that really need a lot of memory to perform their compute. And so we just rolled this out. We got this cluster in April of 2021, and we've got a bunch of users using it for all kinds of things. My lab's using it. Chindo is a co-PI on this grant. His lab is using it. Also, Carmen Canavier is using it for some computational neurobiology. And in the interest of time, I'm not going to say much about this because Chindo covered the master's program in bioinformatics that's is being initiated at LSU Health Sciences, and you've already got the link to that bioinformatics program. And lastly, I'll say that I'm one of the founding members of the Microbial Genomics Resource Group at LSU Health Sciences. And so, if you follow that link, it's essentially, it operates sort of like a core, but we're not really a core. So we just call ourselves a resource group. And it consists of myself in charge of the bioinformatics, David Welsh, who's a pulmonary medical doctor, and Mung, who does all of our sequencing prep and sequencing, and then a variety of postdocs and students who work with us. But we have on this website, we have a lot of our joint publications and everything, but down near the bottom and also accessible via this link is our cost and our sample submission. So we do a lot of microbial genomics because we have an Illumina MySeq. And so, we do a lot of microbial community analysis, and we're just starting to get into some shotgun metagenomics as well. And so that's pretty much all I have to say. And, if there's any time for questions, I'm happy to answer any questions.
All right, great. Thank you very much for this wonderful presentation, Dr. Taylor. And we do have a few questions that I think will require a lot of time to answer at this point. We have our next speaker with us already. So what we'll do is we will collect all of the questions that we've had. I mean, we obviously shared the links to all of these programs and labs. We do have a page where all of the speakers are listed out, and links to their websites are there as well. Maybe Ojasvi or Sonalika, somebody could post that link again. With time in mind, Sonalika, maybe we could introduce our next speaker.
Thank you so much, Ilya. Now I would like to welcome Dr. Shamir Kadir. He would be talking on pharma and AI, integration of omics and clinical data. Dr. Shamir Kadir is the Senior Director of Advanced Analytics, Data Science, and Bioinformatics at AstraZeneca, a global science-led biopharmaceuticals company. At AstraZeneca, he leads a global team of data scientists, biomedical scientists, statisticians, and software engineers as well. Over to you, Dr. Shamir.
Thank you. Thank you for the kind introduction. And thanks again, Elia, for hosting me today on this it's Sunday, but I can already see that there are more than 100 attendees in the talk. So I know the science is strong here. Kudos to you and the team for organizing such a wonderful presentation, wonderful meeting. Let me just get this slide shared. Okay, can you see my slide?
Yep, we can see a slide. Great.
I just want to make sure I share the right one. Okay, it should be fine. Okay, you can see my slides, right?
Perfect. Thank you.
Okay. Yeah, thanks again. I'm here to talk about two of my favorite things, like about pharmaceutical industry in general, and also about AI. And then in a sense, what I'm going to do today is kind of give a little bit of a background about why this is important. Why is right now the moment for these two sectors to kind of come together, and with the specific example of how omics data, or for that matter, multi-omics data and clinical data that we typically generate, one will be generated in an experimental setting, whereas the other will be mostly generated in a clinical setting, how and why we need to put them together. So this is more of a pedagogical style talk. The reason is that Elia and the organizers kind of mentioned that there will be like a large number of students attending this meeting. So rather than brand new data, what I'm going to share is some of the published work, and then also show how we are developing some advanced analytic solutions to address some of the current challenges in integrating these types of data using a combination of machine learning and machine intelligence approach. So my group's research focus, we're primarily can be divided into sort of three main themes, like translational bioinformatics, computational medicine, and healthcare data science. But the group, my lead a machine learning research group, and the focus really is to leverage high dimensional multi-scale data to design, develop, and repurpose new therapies. That's the single line of research focus. But how do we do this? We are a computational team. We are from the get-go leverage advanced machine learning approaches. What we really wanted to do is we really want to sort of close the gap between the way we generate data, the way we interpret data. For many of you who are on this call, are very much familiar that the current era of biology, especially I would love to call it digital biology, is moving at a very fast scale and pace. But at the same time, if you go into a hospital, you'll be still measured your blood pressure using a blood pressure cuff that's I don't know 60 or 100 years old, or you're using a stethoscope that's like 100 years old, older technology. But how can we bring these two rather different worlds together? Maybe we could help here as a group of data scientists and machine learning scientists. We could help with the combination of data and technologies. And why data is important. And you might have heard about big data, some call it deep data, but for me, data at any scale or any grade level range of quality is interesting, whether it's big, deep, small, or data crumbs, doesn't matter. As you can see that these are some of the world's most popular magazines, all the way from Fortune to Wired to Popular Science to Economist, and then also two of the leading science journals. They all talk about data can bring in value. But I would tell you that it's not just the data, it's about what question you ask with this and what tools do you use to mine this data. And then we'll see some examples as we go forward today. And let's kind of come back from that, specifically to the setting in biology. Like for example, from if we want to go from a disease, for example, diabetes to a mechanism to a therapy we will bring it to the patient, and then a patient gets a better outcome. Can that drug be working for something else? And a good example would be like a classic example would be metformin, like that. But then what happened is that we're seeing a paradigm shift thanks to both the technology aspects as well as improvement in biological profiling technologies as well. So there are like both the scientific as well as the technology is in a paradigm shift here. So for example, we are moving from a traditional data type to evolving data type. You don't need to have all of your data in one location to do centralized analytics. You can now do decentralized analytics. You can go from GBs or TBs in data to petabytes and exabytes of data. Your data can be structured, can be semi-structured or unstructured. You can use NLP, text mining, and other types of techniques to bring structure to your data. The data model could be a in olden days, like I still remember, like when we do a health informatics project, we need a stable data model. That's gone. That time is gone now. Your data model could be evolving flat data model. data is moving from a low dimensional to high dimensional, and statistical approaches, and hypothesis-based approaches are now kind of moving to more machine learning or deep learning based approaches. I'm not saying one is better than the other, but we can combine these two, and you can see this in studies. For example, let's take the same example of diabetes, type 2 diabetes. You can see that one of the first GWAS had, maybe 1,000 patients, but the last GWAS on diabetes have like 100,000 plus patients in that study. So as you can see, we are in this paradigm shift here. But at the same time, we also changing our data source. Earlier, we used to either a lab will generate a data set, they will analyze that, they'll have an insight. Or maybe we collect studies, like 10 studies that are related, and we do a meta-analysis on that. We don't have to be restricted like that anymore. If you have a good question, you can bring in data from anywhere. But there are like data repositories that are out there. Obviously, some of them are a commercial product, but some of them, like UK Biobank, is completely available for or those who are interested to ask interesting research questions with that type of data. So you have biobanks, you have biorepositories, and then earlier in the days, it was very difficult to get clinical trial data to do reanalysis, but that is also changing. We now have several projects that tries to bring in clinical trial data in an open access way, for example, whether it's ct.gov or a aggregate analysis of ct.gov. So you can see that all of that's coming together. And then not just this, now we can measure, typically, when we run a clinical trial or study, when a patient comes into the hospital, we can measure a few things, and then when they leave the hospital, we won't be able to measure anything anymore. But then that's not the case. We now have sensors and wearables which are affordable and accessible and can be integrated with your study program, and that brings you a variety of data as well. So you just don't have to restrict to just the omic data that you typically measure in a lab setting. You don't have to restrict to a clinical data that you capture in a hospital setting. You can now get into the real world and collect data from there. So that's one of the gist of the changes that we are seeing right now. And this is why this is possible right now, because we see a huge shift in the technology landscape, and a lot of technologies are kind of growing together thanks to access to high performance computing, and whether it's through cloud or local or a hybrid approach, or advances in both the biology as well as technology, like I mentioned, whether it's better natural language processing approaches, like you can mine a large amount of data together. You have more intuitive data models available. You can have a precise integration, governance, and security to ensure that the data is being accessed in a privacy preserved manner. So these are all possible right now, thanks to all of that progress. But when we come to drug discovery, development, and repositioning, what's all possible? So this is just an example of what's possible if we bring in AI into the whole enterprise of developing drugs. So it can definitely help in novel therapeutic agent discovery. You can do precision disease stratification, for example, within a population who are the disease population. Within that disease population, what's the subset of patients that need treatment A versus treatment B? You can do integration of multi-omic data, which we will see today. And then we can also use the same type of technology to understand that, how physicians are utilizing the therapies that we develop. When, like I mentioned, when a patient is not within a hospital, not enrolled in a trial, we can still monitor them if they're, using an approach that's called continuous remote monitoring and diagnostics. And then also we can plan, design, and optimize our trials as, using AI-based approaches. So just to kind of give a broad overview that AI is being applied across this enterprise of drug development. But due to the time constraints, we'll only focus today on the integration of multi-omic data, and we see a few examples on that. And even within this particular focus area, as you can see that there are a lot of possibilities here. But we'll pick 3 examples, like I mentioned. The first 2 are published one, and the last one, it's an ongoing project. I want to share give you an early view into the project, and thanks to our leadership, this is currently being developed as an open access project. We'll be talking more and more about these projects, like these type of community activities, and we'll be happy to share the tool with you all, and would be great to hear from you about the tool in hopefully before end of this fall. That's our plan. Okay, before, so that's sort of a background of my talk. So I'm going to talk about 3 examples today. This is my first example. For those who are new to the whole genomics, clinical genomics-based drug discovery, it is a quick example. You might have heard about loss of function variants. These are genetic variants that could lead to an aberrant RNA product. And what happens is that you will end up with a non-functional protein product. And the concept is there in the literature. We have seen that it's been successfully used to find targets and now being translated to therapies across the industry, that you can see. And then the key 3 approach that loss of function could enable is you can probe biology. You can if there is a loss of function that leads to variant that leads to let's say loss of a functional protein product, would that lead to a particular phenotype? What is its clinical implication? Can we use that approach to develop a therapy? These are the things we can do with the loss of function approach. And there's a lot of research has happened over the last few years. But what we were interested, this is a work that I've done in collaboration with my colleagues from Mount Sinai. The reason was that what we wanted to know was, like, let's say if we have hospital-based clinical data, and then also have a biobank that's linked with clinical and genomic data. So what you have really is genomic and phenomic information. And then, you obviously need, for this type of study, you need an external validation data bank. Let's hold that for a second. And then I also have data from public repositories, whether it's genotype, phenotype, tissue association. And then I can also, if I do identify some significant signal, maybe I can go and test it in an in vitro testing or in vivo testing, actually. So this is in a way one of the workflow that we have leveraged here. So we started with a database that includes demographic data, disease diagnostic, lab test results, drug information, and all that kind of comes from the data warehouse. And then also more than 11,000 patients with their genomic information as well. So the question that we were interested in, that we wanted to understand if there is any loss of function variants. There are a few already that's been reported, but is there anything that we could find from our own? Because our biobank is a little bit unique on its own. So want to understand, if you look into these few cardiometabolic traits, can we identify a few loss of function examples? So we have a lot of challenges with that, because we're really handling here with very messy type of data. There will be incorrect values, medication effects, and we used an approach called computational phenotyping. The long story short, we cleaned the data. The data is now ready for computing. And then, we did a simple modeling approach, gene by trait association, as you can see in the equation. And then, let's say if I'm interested in a loss of function variant in ApoC3, compared to a wild type variant, as you can see that the patients with the loss of function variant have a significantly reduced triglyceride effect. So that's a classic example. Everybody knows that, and it has implications in developing therapies and all. But then we looked at a variety of them. Like we indeed replicate some of the big ones, whether it's PCSK9 or ApoC3, but we also find something new as well. So we basically kind of integrated that data, like the kind of phenotype data, and then did some candidate association. And then what we identified was, we found a particular variant in a gene called DGAT2 could lead to, in a way, significant lowering in glucose levels. So we kind of followed that up with both, experimental studies. And then what happened was that we were able to find one gene, especially based on these approach, with the validation that, so our mouse knockdown study suggests that DGAT2 as a potential target for glucose lowering therapy. And then we have identified a few others as well. Let's say if you're interested in lowering triglycerides, we found something, a few genes including RNM, TL1, SCRN2, PCK2, as potential targets for lower cholesterol. So think about it. This is only a small database, and it's a fairly large EHR data, but the linked data is very small. And UK Biobank has just released with hundreds of thousands of patient sample. So really, I want each and every one of the students in this call to kind of think about what's that most curious question that you want to ask. Data is never a problem. Data is always available. It's really how creative you can be, how curious you can be, And then you can ask these type of questions, and you can take it to either a therapy, a mechanism, or a target. So that's just one of my first example. And then the other potential theme I want to discuss, and this is again, those who have heard about repositioning, this might be a little boring. For those who are hearing this for the first time, repositioning is an approach where we take an approved or a drug in a certain drug or a therapeutic in a certain stage of development. We take that drug, we investigate whether that drug could work for any other indication. And like you can see in literature, a ton of examples like that. And then what we can do is, like, if you could identify that using a drug that's in advanced stages of development, that could potentially be developed into a therapy for a new disease. So we could potentially save some development years, and then also the cost, time, and energy too. But at the same time, you still have to do repositioning is not just a computational part, but you have to do downstream analysis, downstream clinical trial. But the time to trial is where we are going to save, actually, rather than, it's not like when we hear about repositioning, don't think that it's an experimental approach where we do completely in silico. That's not the purpose or the aim of repositioning. You can do that, but really the value will come when you take it to a trial. And then there are a lot of different ways to do repositioning. And one approach that we've been focusing on, this type of a molecular matchmaking approach, using one type of data here. This is primarily transcriptomic data. So we can generate what we call as drug signatures and disease signature. And the idea is that, using statistical learning approaches, can we identify let's say if this is the disease signature for, let's say, disease 2, and I want to find a drug signature that could reverse these red to green, and maybe this is a good drug candidate for this particular disease. So can you do that type of a molecular matchmaking for that? And then we were interested in repositioning and did one of the largest meta-analysis in this space. And what we found, and I want each and every one of you to think about this and tell me your thoughts also. When we looked at around 200 or so, 200 plus repurposed agents across 1,100 diseases, what we found when we did a post hoc analysis, these repurposed, let's say I have drug disease 1 and disease 2, and that can be potentially targeted using drug X, they are more likely to have shared genetic architecture. For 31% of them, more than 31%, we saw that. And then again, around 27%, we saw that those two diseases, when you look into the pathways they share, we saw pathway crosstalks. We quantified them. And then the most interesting part, just like, by integrating the molecular data with EHR data, or clinical data, what we found was, if two diseases are comorbid, we see them most commonly within a patient population, they're more likely to potentially be repurposable. This is not a hard and fast rule for it, but again, there is some evidence that's coming in. These are the areas where you should potentially look when you're planning your repositioning investigation. And why repositioning works? Because one of the potential reason repositioning work is because it's pleiotropy. Pleiotropy is a key biological phenomenon, and where one gene could influence two or more seemingly unrelated phenotypic traits. And that might be the reason where, and then the genes are doing different functions in different settings. So when you put to using, one drug against two diseases, maybe there is a better targeting strategy there. So just for you to think about it. And as an example, so we recently, this is again a work that we've done at Mount Sinai. So here, what we did was in collaboration with my friend and colleague, Kamlesh Yadav, who's now at Texas A&M. And what we did was we're interested to come up with a therapeutic strategy for a rare form of prostate cancer. So we did some public data analysis, identified some gene expression data from GEO, and then we basically run a repositioning algorithm. We were able to identify, some of these drugs. And then what we found was, interestingly, an alkaloid, of course, that naturally long black pepper compound called piperlucamine was doing really good in some of our viability assays. And when we looked into the literature and we saw that, this could be a potential target, because there's already been a ton of data on this in the literature. So we went ahead and built a mouse model for this unique sort of rare cancer type, and was able to show that the drug indeed have an effect as well. So really, this is again one of the example I wanted to share with you. And in a similar way, if you're passionate about a particular disease of interest, I would highly encourage to include repositioning as a potential strategy in your research. So the last part that what I want to cover today is we live in, like we have seen that, like we live in a world of data influx, and we have this challenge of we have more data is being generated, but there is a huge delay in inference from this data. What I meant by that is, let's say my friends and colleagues in microbiome or genomic sciences or or multi-omics group generating these different types of data. They know what their microbial signature is associated with the disease. They know their genetic variants, they know their transcriptomic signals, but they interpret them independently. They never put it together and look biology in a more connected way. But biology actually operated in this level. It's an integrated approach. But what can we do about it? And on top of that, along with omic data, we also have like custom assay data. A company like AstraZeneca, for that matter, any other pharma company will have some custom assays, imaging data, phenotyping data, clinical data, you name it. And then instead of analyzing them independently, can we analyze them together? This is a question that's been, really been challenging for us, and just like the entire industry here. And when we started our group, one of the first group that reached out to us, our microbial sciences group, reached out to us and asked, hey, Shamir, can you and your team help anything with this? And I was like really happy, because hey, there's going to be a ton of data on this. We can apply some deep learning approach. And then what I realized was, interestingly, the sample numbers are always very, very small. And then you have this classic, analytical challenge where you have small n and a large p. And then what happened? And then we did an extensive search and literature survey and identified that, why we cannot really use single-omic techniques and then, just make a bigger or better version of that. But they do not take, statistically into the, integration aspect. And then they do not find relationship between the omics. So what we realized was maybe we need to make a wish list of these omics approaches, and then potentially come up with a way that we could, then use to, integrate, analyze, and interpret this data set. So what we thought was, okay, let's write our wish list here. So we started with this sort of wish list, and what we found was there are 3 key ways that you can do omic data integration. You can do an early integration, you can do an intermediate integration, or you can do a late integration. But then what happens was that it doesn't matter what type of integration you choose. We also want to make sure that the data represented in each of these, data or omic or non-omic type should be equivalent. One omic should not dominate. You can still weight your omics if you want, and they should be generalizable. It should not be only work for transcriptomic or NGS-based techniques, or it should only work for imaging-based techniques. And then we need to do variable selection on each block, and then in a way that we have to also do predictive modeling on top of that. And two of the key part was, like, you have to understand the relationship between in different types of omics, and then also it should not be confined to only omic data, but other types of data should also be able to add. And then, we came up with this approach. We did an extensive literature survey, and then instead of developing something brand new, what we thought was we start with a pretty decent package, and then add features that doesn't exist in it. So we basically start with something called Diabolos. As you can see that we have some objective and subjective classification approach that we took here. And then, we identified certain algorithms that could go in there. And then with that, on top of that, we have built some additional layers as well. So these are the key features. We can do a lot of normalizing and scaling within omics world. You can do single omic multivariate modeling. You can do multi-omic multivariate modeling. We've also come up with a new strategy. Let's say you've done all these analyses, then you can actually do what we call as a block rank and identify the best features from that and best model and most stable model, and then you can go do downstream experimental validation as well. So this is, in a way, I don't have time to go through everything, but what I can do is, this is a sort of a broad overview. As you can see here, we have a lot of different types of omic data can be onboarded once you do pre-processing, and then you can do 2 types of analysis, or rather 3 types of analysis. You can do, if you don't know the class label, you don't have a class label, it's fine. You can do unsupervised exploration. If you understand what's your classes, you can do supervised exploration and go all the way to the modeling part. If you want to do cross data set prediction, you can build models, whether it's random forest or support vector neural network, you can do that. So, and then in addition to that, we have other models that we're building towards, understanding the gene sets and repositioning and all of that as well. So this is, in a way, what we want to do here really is to address the challenge in this biological data interpretation problem. So the features are interpretable. 3 things if you have to take away from my talk. So interpretable features, relationship between omic types, it can be inferred, and it's scalable to add new omic types. We have so far done integrated with 12 types of data, and we strongly believe that you can add more types of data as well. So this is a quick example, like this is the dummy data sets that we have put together. So you can see we have control seropositive and disease for a cohort. And then this is our initial model. We do model selection and tuning. Models get better, do better separation. The first one is using this one is using mono-omic data, and this is using multi-omic data. You can see that. And in addition to that, we have an array of novel visualization approaches. You can also understand the stability of these models as well here. And then this is what's available. We will be sharing this with the community very soon, and watch this space. And with that, I would like to come to the summary. I strongly believe, biomedical healthcare data science it can actually optimize, accelerate, reduce the cost of development. As you've seen a lot of examples today, and I'm sure you're hearing a lot more examples throughout this symposium. And as data scientists, be ready to push the limit of your comfort zone. It's a team sport, right? Like we all need different types of idea to push the limits, because we don't know what we're going to find. And that's the joy of being a data scientist. And then the more creative you are, the good handle of subject matter expertise that you have, you can definitely bring in a lot more value. And then, definitely in biology, everything is connected. It's important to ask bigger and bolder questions. And one more thing, we are always hiring, always looking for collaborators, and will be, potentially looking for interns and apprenticeship positions and all, looking to fill all of that. So please be in touch. You can find me on LinkedIn or Twitter, and we'll be in touch if you would like to connect, and we'll be happy to stay in touch. And I would like to acknowledge my team from AstraZeneca, and for the other 2 examples, the team from Mount Sinai. And with that, I'll be happy to take any questions. Thank you.
Great. Well, thank you very much for this great talk. And a couple of questions. So first of all, a personal question. How did you personally end up at AstraZeneca? And can you briefly describe your path?
Oh, sure. Yeah. So, in a way, I wanted to be a software engineer when I was, from straight out of my high school. And thank you for asking that question. So, but I took biology for my high school. So in India, I'm originally from India. In India, you cannot really go to computer science with a biology background. So you have to do some kind of an undergrad in non-biology background and then go to computer science. So, and then I went to an undergrad in physics. And then when I was doing physics, second year or third year of physics, I heard that there is this subject called bioinformatics that combines computer science and medicine. I'm like, wow, that's fascinating. That's really, really exciting thing. And then I wanted to get into a master's in bioinformatics. So instead of going to computer science, I completed my master's in bioinformatics, and I did my grad school from India under Professor Sadamni, like National Center for Biological Sciences. And I worked primarily on building machine learning models to understand protein sequence structure and interactions. And then while I did that work, I wanted to kind of get experience with other type of data. And that prompted me to go to Mayo Clinic for my postdoc. And I was trained with this whole EHR-based data, genomic data. It was in the beginning of the whole sort of genomic medicine, precision medicine phase in the US. We were one of the first site for a project called Electronic Medical Records in Genomics, like 12 years ago, and was part of that, participated in several clinical trials, and then eventually switched to industry. At a certain point, been doing some consulting and eventually switched to industry. And I'm with AZ for now 3 years. The reason I joined AZ 3 years ago, AZ was starting the data science group, and we were like only maybe like, you know, my including myself and my boss, we were like 3 members. Now we are around 60. And now the whole data science and AI is actually 150 plus in AstraZeneca. So we have grown a lot, and really excited to bring, you know, to see the power of, you know, data science and AI in drug discovery. So this, you know, I still remember, you know, in one of the early days of my master's in bioinformatics, I hear that we are going to see precision medicine. Every hospital will have a bioinformatics team, which is something I've seen happening right now. You can see that NGS is in every hospital. I mean, like in one way or the other, the physicians are using that in like one way or the other. So we are really there. I mean, like this is our moment, and yeah, so that's the long story in a short way. Thank you.
All right, great. Thank you for that. Okay, so we do have another question. You mentioned that you did your postdoc at Mayo Clinic, and how different is it working kind of in basic research as opposed to, you know, clinical type information? I mean, how different is the thinking and, you know, the need there in terms of omics data?
So definitely, like, you know, compared to the basic science group where we primarily work with, you know, genes, proteins, and computers. That's where we were working on. Here you have patient data too, but at the same time, you have to think about what is patient data. In the end, what you capture in an EHR data is nothing but, think of it like a large, long experiment, right? Like, you know, you're measuring biomarkers, you're measuring imaging data, you're measuring other types, like, you know, medication data. These are all different types of data that you measure from your patient cohort. That's being compiled into, you know, not always a user-friendly database. You always need to do a lot of processing because there's a lot of human errors, because this will be either typed in or translated by a, you know, an automated transcription engine. You have a lot of clinical notes that you can mine through. So there are a lot of that pre-processing is required. But in the end, the way that I thought about that was, you know, this is pretty much like an experimental data, but it's coming from, you know, a physician and patient encounter, you know, and that's how I kind of thought through that. And now what we're seeing that we, you know, when we were back at Mayo, the challenge was to put genomic data into electronic health record. We have achieved that, you know, it's there and everybody is aware, and we still need more training. We still need, the accessibility is not global. We need to improve that. All of that is for true. But now the field of the experimental biology is moving at much faster pace. And as you can see that, like I mentioned, biology is now more digital biology, and we have that gap. They're generating more and more data, but we are not able to interpret them on time. So how do we do that? And that's where approaches like Omics fold, and, you know, these kind of strategies and needed. And again, we cannot do it by ourselves. We need collaborations. We need to work with the community, and I really appreciate this invitation to kind of share our research with you all. Yeah.
Great, thank you. Another question. So while recruiting for bioinformatic roles in AstraZeneca, is a PhD an advantage, or should you have only a master's degree?
We have, you know, I can speak for the group that I am part of, like, we are data science and artificial intelligence. So we have a senior data scientist, principal data scientist, and then assistant director, a director, senior director, and then executive director, all the way up to the VP. So that's sort of our career level in data science. We don't have an absolute requirement for a PhD. So if you're very good at, you know, in some of the technical skills or scientific skills, have, you know, a passion to work on solving the type of problems that are related to drug discovery, development, and repositioning, or even the entire enterprise of drug development, including clinical trials or patient recruitment. We hire, you know, both PhDs and masters. It's not just, you know, even we have undergrads too, like a very few undergrads too.
Okay, perfect. So we are going to be starting our panel here in just a few minutes. Right before that, and thank you, Dr. Kader, for your insights. We're going to continue with the panel, so please stay here. But before we jump into that, we want to briefly summarize what we heard up until now. We have just a couple of minutes to do that. So let me do that. I'm gonna share my screen. And I did want to mention that Dr. Kader mentioned Omics Fold. Just as you were speaking, we posted a link to the GitHub repository. I don't know if you have any other links you would like to share. You know, maybe you could post them in the chat and our team will make it available to everyone. And I will just briefly again summarize here what we have heard from all the speakers up until now, and then I'll pass it on to Mohit to introduce the panelists. So the first speaker that we had was Professor Seema Bhatnagar from Amity University, the Assistant Director of the Biotechnology Program. She really talked about her research in India in precision medicine in cancer, some of the work that her group is doing on computational analysis of different proteins and interaction with different precision treatment drugs to avoid adverse effects of other treatments. But she also spoke about the biotechnology program that includes bioinformatics at Amity University and how students can get involved, especially the programs in India, but also international programs for international students with the possibility of also attending the campus in the US. So that was a great talk that we heard first. We then continued on to talk about a different type of a program, which is a program for master degree students. Professor Sona Vasudevan, the Director of Systems Medicine Program at Georgetown University Medical Center in Washington, DC. Some of the things that they are doing in research in terms of systems medicine. So obviously, what we just heard from Dr. Kader as well, basic research, biotechnology programs, and then medical challenges and kind of issues that students have to face. She also talked about the kind of sustained interest and increased interest in machine learning and how we are working together on addressing that problem in her program for the students. We then we heard from Dr. Philip Kumpel, who's the Associate Teaching Professor and Assistant Department Head at Carnegie Mellon University, one of the most exclusive programs for computational biology in the country here in the US. Very difficult to get into, but a lot of opportunities for those that are persistent and are really looking to acquire coding, computational, algorithmic development kind of skills. So again, a very different flavor, again, of bioinformatics and computational biology, but very important to kind of understand what are the perspectives and opportunities. They also have high school programs for high school students to have a summer experience, understanding how algorithms can solve real biological challenges, and some of the interesting projects that they're doing in microbiome, environmental microbiome, as well as kind of interesting work that they're doing on COVID-19. So then we went on to hear a message from Dr. Gus Kasoulis. He is the department head at Louisiana State University, but also the PI of Louisiana Biomedical Research Network and the president of the National Association of IDEA Principal Investigator that really brings together 23 states and Puerto Rico. It is hosted in Baton Rouge in Louisiana, where his primary role is, but he is essentially responsible for kind of overseeing the implementation of some of the IDEA programs around the country, helping elevate biomedical research and providing opportunities for students, over 50,000 students, to learn about bioinformatics, learn about data sciences, which is, of course, one of the mandates for NIH and NIGMS, which is funding this whole IDEA program with over 300 million in research funds. We then heard from Dr. Chindo Hicks, who is a professor and program director at the Department of Genetics at LSU Health Sciences, which is a kind of a center focused on genetics, but also working on multi-omics integration, working on health disparities. And one of the highlights, I think, from his talk was the interactions that they have with international students, especially with programs in Africa. And so we have a lot of students from African countries that are looking to kind of continue their studies, continue to PhD programs, master's programs. They do have a master's program here, but they also do a lot of collaborative research. And so I think a great opportunity to hear from them about that program. We then heard from Dr. Chris Taylor, who is a professor of microbiology, immunology, and parasitology, and the director of bioinformatics, biostatistics, and computational biology core for the Louisiana Biomedical Research Network. And, he shared a few very interesting aspects of his work directly on microbiology related metagenomics analysis. As his background is also in computer science and kind of coming into bioinformatics from that perspective, he also provided interesting insights into the needs for high performance computing in a lot of these larger studies and some of the exciting projects that they're working on related to precision medicine and related to also training and education that is being made available widely through the Louisiana Biomedical Research Network and directly from LSU Health Sciences Center. And then finally, we heard a great talk from Dr. Kader. He's the Senior Director of Advanced Analytics, Data Science, and Bioinformatics at AstraZeneca. So really interesting, I think, overview of computer science oriented programs, medical type programs, and then basic research programs around different countries, around different locations in the US. Last day, in the previous day on Saturday, we heard about other countries. And we did hear today from Amity University, which is in India. But I think this talk brings us to the topic of our panel discussion and really highlights some of the important similarities and differences in the way that bioinformatics can be used across a variety of different domains. And without much further information on what we've heard, I think all of these talks were really exciting and a lot of information for us to digest. But at this point, I would like to pass this on to Dr. Mohit Mazumdar, who is going to be introducing the panel speakers for today. And I do want to mention before he does that, that we do have a full list of speakers with the links that we have received from them. You can find all of this information on this link. 1 second, I will put it for everyone. Okay, so if you go to this link, you will be able to find all of the speakers from yesterday and today and the links to kind of learn more about all of that. So over to you, Dr. Mazumdar.
Thank you, Pia. Now, I would like to welcome everyone to this much awaited panel discussion focused on bioinformatics in industry. So today we are privileged to have some of the leaders from bioinformatics and biomedical industry involved in pharmaceutical industry, assisting in modern drug discovery. So our first panelist, Dr. Samir Kadir, is a Senior Director of Advanced Analytical Data Science and Bioinformatics at AstraZeneca, a global science-led biopharmaceutical company. So at AstraZeneca, he leads a global team of data scientists, biomedical scientists, statisticians, and software engineers. He oversees profiles and portfolios and projects in oncology and biopharma to accelerate drug discovery and development using biomedical and clinical data and machine learning approaches. So we just heard from Dr. Shamir sharing insights from pharmaceutical perspective in applying bioinformatics, especially putting emphasis on data and the technology, applying data science and envisioning artificial intelligence in the pharmaceutical industry. Along with Dr. Shamir, we have Mr. Tarun Walia, currently at MIT, selected as prestigious Sloan Fellow. Mr. Tarun is a biotechnologist by training and with a broad experience in areas such as biotechnology consultation over 14 years, and then worked across 3 different continents. He is an expert in business strategy, R&D planning, and pipeline strategies. His work also involved in marketing and branding strategies in pharma and biotechnology. Our next panelist is Mr. Anthony Finbo. So Anthony Finbo is the CEO of Eagle Genomics and has achieved success over many years as CEO of technology businesses. He has occupied every seat at the board table, chairman, CEO, CFO, NXT, and investor director for both public and private companies, and now assists leaders and their management teams in meeting and overcoming the most significant challenges that they might encounter on their journey. In the panel, Mr. Finbo will share his perspective on different aspects of the gap that he sees in academia and industry in bioinformatics, and the vision he has built to build an ecosystem with students, faculty, partners around the world to advance the bioinformatics or biotechnology platforms that enables Eagle Genomics as a company. Next, it's an absolute pleasure to introduce Mr. Nahid Kurji. Nahid Kurji is the president and CEO of Cyclica, a Toronto-based biotechnology company that leverages artificial intelligence, computational biophysics to reshape the drug discovery process. So Cyclica provides the pharmaceutical industry with an integrated, holistic, and end-to-end platform that enhances how scientists design, screen, and personalize medicines for patients, and has recently been named by Deep Knowledge Analytics as top 20 AI in pharma companies globally. Nahid is also co-founder of the Alliance of Artificial Intelligence in Healthcare, AAIH, of which Nahid is one of the 4 appointed executive officers. So he has been featured in Global Mail, Forbes, Vanity, Wuxi Insider, and has been invited faculty in many universities discussing topics such as AI in medicine. So I welcome you all here. And before we go into the actual discussion and begin the session, I would like to give everyone maybe a brief overview of how things have shaped up and why we think this session is so important. So from genetics and toxicology to microbiology to radiology, there are, different branches of biology to specialize in. And out of many, bioinformatics is one of the intriguing fields which enables us to identify, evaluate, store, and retrieve biological information, right? Something that we have learned throughout this Omics Symposium. Now, integrating the elements of data science. So when it comes to medical research or product development, data science refers to a broad, rapidly evolving field that supports the analytical process of extracting knowledge from a variety of data sources and types, right? And then integrating data types and resources such as omics data types like genomics, transcriptomics, proteomics, phenomics, images and wearables the data that we get from there. All of this this gives us an holistic understanding and identifying the most meaningful interferences that leads to the best decisions. So big data in biotechnology in today's world is not just only about genomics, but it's also about the data that may be collected by sensors, wearables, ingestibles, or implantable sensors that can provide this continuous data stream for clinical health, clinical trials. So in biopharma industry, the focus has now shifted towards processing data more effectively and deriving relevant insights to realize its full potential. So by working together, different analytical methods offer this holistic pictures and translate this into practical steps for product development, maybe marketing or maintenance strategies. So with this overview, I would let the panelists to take this forward and share some insights on how the future bioinformatics and data science that they envision and that they see with experience, and the best approaches that you may have figured out from your own experiences. So I would like to invite first Dr. Shamir to share his thoughts on this.
Thank you, Mohit. Really appreciate that great background and great overview. And thank you, fellow co-panelists. And in my opinion, we're seeing a sort of a transition in our industry, especially in bioinformatics industry. And I'm also a little bit concerned about that too, because everybody now calling it, data science, and then, either biological data science or biomedical data science. And we have seen that also, we have seen that even in the evolution of bioinformatics, we have seen that too, it has gone through certain cycles. But now, we are in a stage where, a bioinformatics person should have definitely the domain knowledge, domain expertise. He should know his tools, databases, and all. But on top of that, having that edge in AI, machine learning, and predictive modeling are going to give them, better access to jobs, positions, and internships, in my opinion. So that's where I'm thinking. And then the other potential observation that I have is that the data science on a broad is there was a Harvard Business Review piece saying that it's one of the hardest career of the century. But then again, so calling it a biomedical data science is actually good. But remember, you have a particular edge because you are trained in this domain from the get-go. So that would be my thought. I would invite Anthony to kind of comment on his observation in this theme.
Thanks, Shamir. Thanks, Mohit. And also echoing Shamir's comments, delighted to be participating alongside such a deeply competent panel here, amongst whom I'd say I'm the interloper, because I didn't and don't have a background in bioinformatics. I'm a serial entrepreneur. I'm an interloper, but I can claim some substance and knowledge because 30 plus years ago, I did my studies in cybernetics, control engineering, and maths. And that's deeply information theory related. And I have to say, I was delighted to discover only quite recently that Claude Shannon, sort of godfather of information science, I discovered he did his PhD in genomics. I don't know how many of us knew that, but it was a delight for me. I felt slightly less of an imposter. But I come at this from a very different perspective. So look, I'm an entrepreneur. I've built 4 or 5 companies over the years, and in a number of industries. And I've either listed those on the London Stock Exchange or the NASDAQ, or sold them to US NASDAQ listed buyers. And I entered this industry about 5 years ago. And Mohit, I'm just fascinated to see how radically understated the size of the market you projected there on your initial slides is. I mean, I actually came to this space because of the tremendous innovation opportunities. And, I'm just following most recently the guidance provided by McKinsey & Company, and by Boston Consulting Group Henderson Institute, the research establishments that those consultancy firms have developed towards Industry 4.0, which it's increasingly clear is going to have a very significant bio dimension to it. McKinsey & Company describe it as the bio revolution. And to participate in the bio revolution, Boston Consulting Group Henderson Institute describe the opportunity as nature co-design. And so, my perspective on the market is very much bigger than just drug discovery. It is application of bioinformatics and data science technologies to enable customers to participate in the biorevolution. And to that end, we at Eagle Genomics are building a company, a platform company, to enable microbiome science, networking of microbiome science in the first instance, because we perceive that to be the most proximate battleground, if you like, in the biorevolution. Now, just particularly to the question, I think a lot of my frame has been shaped by what I've done over the last 20 years in telecommunications industry, network software for telecommunications industry. 20 plus years ago, that industry was radically disrupted by the introduction of TCP/IP and Internet Protocol. Before then, it was time division multiplex networks, and the 2-bit code Internet Protocol disrupted that industry, first slowly, then incredibly fast. And what happened in the early stages of that disruption was that big companies, the big incumbent telco companies, tried to build groups of people that understood internet protocol and could build the technologies to exploit it. And that's what I call first generation dynamics in these enterprises. As the market evolved, we started to see best-in-class applications coming from external vendors and more of a focus on the underlying business from the telco operators. And of course, our industry here, the industry we're all in some way participating in, is being disrupted by the 4-bit code, DNA. And first, in the early stages, we're seeing a lot of companies experimenting, evolving their capabilities to exploit that understanding of DNA, human, animal, microorganism, et cetera, et cetera. But I think what's clear to me as an outsider is that, and I'm not alone in this observation, the dirty secret of data science 1.0 is it's not scaling and it's not scalable. And in order for the industry to really evolve, data science 2.0 is required. And that's a kind of blend of informatics and data science skills with enterprise class technology. And so what I'm trying to do and what we're trying to do is fuse enterprise class software skills with bioinformatics skills, data science skills to create this opportunity to scale applications that we can promote to large enterprises that want to participate in the bio revolution, but also to network research establishments so that we can enable collaboration between the players in this bio revolution. And I think that's liberating tremendous opportunity for people in the bioinformatics industry. I think the bioinformatics career path is evolving really, really quickly. I think it has to have, and it's incumbent on us to infuse entrepreneurial skills as well as technology skills in the students and the researchers that we're cultivating for this bio revolution. And I see nothing but just incredible opportunity in that regard.
Thank you, Dr. Anthony, and thank you, Mohit and Shamir. I can go next. I'm Tarun Walia. I'm a biotechnologist by training, as Mohit mentioned. And for the last 15 years, I've been working at the cusp of biotechnology, commercialization, as well as data science. And I agree with a lot of thoughts that you both have already put forward. And I believe the opportunity is high. There are a few things that I really advise a lot of people to focus on is, going multidimensional. But what I mean by that is, for years, the data science has been studied in a very different way from what biomedical or pharma or discovery has been studied in. Now, bioinformatics is a field that, of course, ties up the two, but there are still so many gaps which are there and which, you come to know when you're speaking to a core data scientist who's doing machine learning or building AI models. And when you talk to a research scientist and he's more willing to do those mouse models and the research, right? So how do you tie up the two? Is there a way where you can, if not an expert, is there a way you can understand both areas really well and then tie it up with the commercialization process? By commercialization, I mean is where do we have a window of opportunity for a drug therapy to be there or a diagnostic to be there? So just having the commercial acumen tied up with the data scientist and tied up with the research side is something that I really want people to focus on in the future.
And I will finish off with comments. So Naheed Khurji, it's such a pleasure to be here today and to listen to and join this panel. I'm also not a scientist. So Anthony, there's two of us, and I'll consider myself an interloper as well. And my background is historically in finance and strategy. And back in the day, I spent a lot of time in humanitarian aid before then coming back and making a contribution back to the life sciences, which is where I believe the most impact can be had in humanitarian work is how do we make medicines and make those medicines available broadly to people all over the world. And how do we think about data, balanced data, fair data, the availability and the application of that data in the pursuit of medicines to cure diseases, many of which are so rampant in areas of the world where there are patients who may not be able to afford the prices that exist in Western or more European societies, but still have desperate needs. And so that's where I spend a lot of my time. And it's a pleasure to be here today. When I saw your opening remarks, Mohit, in that one slide where he just showed the growth of, I think it was data, it reminded me vividly of the growth of the protein data bank. Now, a little bit about Cyclica. We are a quote unquote AI for drug discovery company. And in today's environment, we design small molecules to interact with desirable proteins and avoid undesirable anti-targets or proteins that you'd want to avoid for toxicity reasons or for selectivity reasons. And so for us right now, as a 60+ person company with offices across the world, our strength on the R&D side is in structural biology, computational structural biology, computational biophysics. And if you go back to Linus Pauling and the determination of the first protein structure, I think that took him approximately 22 years to deposit that first protein structure into the PD, what is now the PDB, an open source repository of all protein structures. I think that was back in 1948, and it took him a lot of time. And then in 1978, the PDB became available. And if we look at from 1978 to about 2013, when Cyclica was formed, there was a fairly exponential growth in the total number of protein structures that were available in the PDB. When we started, there were about 95,000 protein structures that represented, give or take, 20 to 30% of the entire proteome, the 30% of the genome. Today, 181,000. In a span of 7 years, it's doubled. There's more structures being deposited, increasing our knowledge of biology, increase our knowledge and availability of things to prosecute. And when you couple that, which I will say is an asterisk, that was before AlphaFold, DeepMind released their AlphaFold 2 code and have now made available essentially the entire proteome across 20 species, and over the next few years, hundreds to thousands of species. Before all that, there was this exponential growth of data. So when you take the availability of proteomics data, genomics data, transcriptomics data, clinical trials and real world evidence data, you have amassed an availability unlike any other time in history on data. But the most important thing that we've been thinking about is not the amount of data, but the quality of data, the representation and the fairness of data, and then also how to use that data in a thoughtful way to solve real problems, not just to take data to solve any problem. And so for us, it's abstracting the problems that exist in biomedical science, truly understanding bifurcating it, truly understanding the problems that exist, and then matching the data with the algorithms, and then with the advance of cloud computing, whether it be GCP, Amazon, Azure, and now with NVIDIA and how strong they're getting into the biomedical science space with GPUs. And then what's coming down the pipeline of quantum computing. And so for us, it's how do we take all of this available data, take cloud computing, take algorithmic development, whether it be machine learning or deep learning, and start asking ourselves questions of what's possible and what are the problems that can be solved. And so for me, as a non-scientist, by classical training, I've spent an inordinate amount of my time studying and spending a lot of time with people at the interface of those three areas. And at Cyclica, when we look at the team of students that we have brought in, some straight out of bachelor's degrees, MSCs, PhDs, postdocs, people from industry, we're living within the intersection of experimental science, computational science, and then humanities. Do they have the deep empathy to go and seek out those problems in a very new way? And so I'm happy to kind of talk a little bit about how we've built a team. We're going from 60 to about 150 over the next couple of years, and what types of skills and experiences we are looking for, and what I encourage students to go seek in the pursuit of what is the biggest opportunity, which is the making of medicines.
Thank you, everyone. So really good insights. And I wanted to jump in here and kind of, direct the conversation towards, first of all, I will start from something that Dr. Khader already mentioned. As a biology interested student going into computer science and then finding himself at the end in this career of bioinformatics or data science, And kind of picking up on some of the other highlights that all of you agreed on, that really industry is not just solving any problem, like Nahid said, but specific problems that are, the real quote unquote problems to be solved, right? So there definitely exists kind of like a gap, I think, from a student perspective on what does that look like, right? And so hearing about all of these exciting topics, addressing medical needs, bringing better medicines to market, maybe solving some real scientific challenges. How do you envision somebody that is still a student or an early career scientist kind of understanding what exactly, when you envision this big machine of 150 people working on a particular problem, or, thousands of people working on another problem, how can they kind of see their future using bioinformatics in some of the problems that your companies are working on? So maybe I can start with Anthony. What do you think about that?
Well, it's each and every company is, despite the opportunity we all have to research at our desks, every company is by and large a black box, and you can only glean so much about a company from the outside. And I think it's incumbent on us to therefore provide structured paths for individuals that can know only so much, that are competent, capable, that are increasingly sought after resources. It's incumbent on us to create the pathways for them to explore and then specialize, after a period of time within the organizations that we're creating. And I think, by the way, another thing I'd say about myself just quickly is that I left school and did an aircraft engineering apprenticeship. I ended up fulfilling my boyhood dreams really early as a radio radar engineer on Concorde, the supersonic airliner. And then I got fired because British Airways, which was the company I was working with, got privatized. But I look back to those 4 years where I just learned horizontally across a huge number of domains before I started to specialize, as formative in my career as an entrepreneur and as someone who's able to do things. And so we're trying to instantiate that apprenticeship structure, apprenticeship program within Eagle Genomics. We're working closely with universities in Cambridge to develop and deliver what's described over here in the UK, at least, as a level 5 apprenticeship, so that people can explore, they can attach to individuals that are experts in a range of domains. They can be mentored by these people, they can discover their core skills, and they can recognize where their strengths lie, and therefore where they ought to put their energies, because we're great believers in strengths-based coaching, identifying what people are cut out to do unconsciously without being burdened by doing it, and then vectoring them towards that path so that they can really exploit their skills. I think that's one of the things I'd say. The other thing I'd say is that the field is moving so incredibly fast that one has to look forward as well as backwards. When I talk about backwards, I mean the technologies that are available today to learn. They are going to be evolving so quickly that one shouldn't be worried too much about specializing, and one should be open-minded about how to evolve in the future. What I'd say also about the focus we have is that we're in much the same way as Nahid has described his business, we are trying to network microbiome science. So we're using network science as the kind of fundament on which to build our platform and our business. Delighted to say we raised Series A funding last year, and we managed to get one of the leading thinkers on network science, Laszlo Barabashi, to invest in our business. We're about to raise a Series B round of funding. But I think what I'm trying to describe is a picture that's highly fluid in the early stages, so that we can attract the right resources, provide them with a framework such that they can learn broadly and then specialize deeply. And I think we have to offer that kind of career path to individuals, lest we try to get people too focused too quickly.
Great, thank you. So Dr. Cotter, would you like to add to that from your perspective?
Sure.
Thank you. Yeah, some interesting statistics I want to share with this group. Actually, over the last year, I might have interviewed more than 100, quote unquote data scientists, sort of junior to senior level role, and then also been leading some of our internship and program, global internship program, and we recruited more than 15 interns over the last 6 months. But we received more than 800 applications, right? Like you just, just think about it, like, and then for each and every one of you who are students and trainees in this group, how can you differentiate? And this is just, one company's one department, looking to fill a few internship positions. And that particular position, the one where we got 800 was actually for a US based. Unfortunately, this was only open for US based graduate students, so there are like certain country requirements for some of these positions. But we received applications from everywhere in the world, which is okay. Now, unfortunately, we couldn't hire all of them. And then and then when I was going through that whole process, whether pre-screening, screening, and then finally interviewing a lot of candidates, we finally hired our interns. Actually, they're about to finish their projects and all. What I wanted to kind of tell you is that what was that quality that led to that final offer, from my perspective, right? Like they were, doesn't matter, in an interview, and that's a traditional interview process where you send an application, you review the CV, you bring them in, and then you have a face-to-face interview. But then that first step is to select your CV. So it's definitely important what you put in your CV. So if you're a student, make sure that you have some kind of a project-based learning. Whether it's putting extra time with your professor, or collaborating with another friend, or working at a company that's straight across your university, go ahead and do that. That's as important as your coursework. I know schedule is tough, and right now we live in a pandemic world. We cannot really go in IRL. That's not possible. But then you can reach out to them, send an email, make sure that you have that. So one of the factor that stood out for these applicants were they had some kind of experience. And then the question would come from students, oh, that means we always have to have a project to get to an internship. No, that's not the case. You can still be doing undergrad in anthropology and want to do human data science. That differentiate you, just like what Nahid was mentioning, right? Like you bring in a different perspective to the group. That's also okay. And then the other part is when you come into the interview, it's your communication and clarity. Doesn't matter whether you worked on, I don't know, the predicting seed properties, or you worked in identifying how to launch satellites. It doesn't matter unless and until you are able to give your elevator pitch in like 3 minutes or so. We all have, if you take humanity itself, our attention span is reducing, right? Like we all have that issue. If you think about the most leading social media platform right now is TikTok, an average length of a TikTok video is 15 seconds. So we are living in that world. So you have to have that, your elevator pitch ready. You have to have clarity and communication. And then, let's say if we hire you, what matters the most? It's not, maybe it doesn't matter whether you are one of the world's best machine learning scientists or programmer. These skills that we talked about, you can communicate clearly. You can have good engagement with your stakeholders in an industry perspective, whether it's your group that you work with, your line manager, or the team that you're working with. And then also making sure that you understand the change management aspects that you will bring into the table. What I meant by that is, whether it's, stakeholder engagement and change management, these might be new to many students here, because traditionally we don't teach management or strategy to our science STEM-based, STE-based programs. Of course, if it's mathematics or management, depends on that. But stakeholder engagement is how do you deliver your project in a refined and in a sort of a very much in a predefined way, and make sure that all the deliverables are met within your schedule. And then you need to have some kind of a direct communication line with all your stakeholders of the project who either funded your position or funded that project. And then what's change management? Let's say you're building this beautiful algorithm, whether it's for small molecule prediction or to identify a cancer patient subtype. Who's going to use that algorithm? Will they use that algorithm? Are they going to trust in that algorithm? What baseline did you use? Did you communicate that, of course, with the stakeholders, but also with your users? If you don't have a user, a multi-million dollar investment in AI is not going to have any benefit. So these are the things you have to think about, right? Like, of course, your science and technology skills, your scientific and technology skills are important. I'm not saying that they are not important, but are you able to communicate your idea clearly? Are you ready to engage with your stakeholders? Are you ready to have this dialogue to do change management? These are important skills. And this kind of carry forward, it doesn't matter whether you're an intern, you're coming as for a full-time role, or you in your leadership journey throughout this segment of this particular industry. This matters a lot. So the long story short version of that is, to make sure your communication and clarity, that's going to be the number one skill. It's not your Python programming or how to run single cell sequencing. I mean, like, they're good, they're great, but are you able to interpret and explain your result to your lab, to your group, to your collaborators? That matters a lot.
Great.
Do you mind if I jump in and just provide a perspective from a small budding company, not an AZ company? I agree a lot with what Shamir said in overall remarks, but there's a couple of nuances as students are thinking about applying to a earlier stage company than a large pharma. And I will say AZ and Cyclica have done a lot of work with AstraZeneca, and Paul Agapau is a close friend, and Shamir, I'm sure in the data science group, you've worked with Paul, and we've done some and published some work with AZ in the data science space. But here's what I'd like to say to students who are thinking about applying to earlier stage companies. Generally, when you're applying to an earlier stage company, a seed stage, typically 5 to 7, 8 people, Series A, 8 to 20, 30, 40 people, and then beyond, depending, especially in the biotech stage, that's usually where they line up. If you're applying earlier on, I would highly encourage you to truly know the entrepreneur or the co-founders of the company, listen to and read what they've been saying in the marketplace, and understand what that company is talking about, and match your views and visions to that, and what can you do to augment. Because there is going to be, whether you're a co-op, an intern or a co-op, an employee and a contributor going into a management role, that becomes a very important role for that company. And of a group of dozens to hundreds of applicants, they're going to invest a lot of time reading through each one. At Cyclica, at each stage, we've done HR with students well at some points in time. We've done it poorly at some points in time. But what we've continued to invest in is the student marketplace. We have rolling co-ops with the University of Waterloo, University of Toronto. We brought in co-ops from Northeastern University, where we have an office in Boston. That's something that we pride ourselves heavily in. And then we've continued to disseminate content through what was previously called AI Ate My Homework, an online forum where we talk about everything from target selection to clinical development and how AI is playing a role. And we'll have our team and then external folks come and talk about that, generally made available to high school and university and graduate students. And so this becomes really important. But what I'd encourage, as you learn, there's all this material that startup companies disseminate to the market to build their brand. Don't make it an application that is cookie cutter, because that company is differentiated. They're fighting every single day to differentiate themselves. So it behooves you as an applicant to say how you're differentiating yourself and how you and that company are going to help differentiate. And so I've seen far too many just, application that you can take out the name of Cyclica and replace it with any other company. And that loses traction in our eyes, because we are going to, in the information age of 15-second TikTok videos, we fight against that fallacy. And we instead encourage all of our hiring managers to read everything. And as we go through reading that information, it becomes abundantly clear those who are just applying because there's a job application versus those who are genuinely interested in earnestly joining Cyclica. And the last thing I would say is that there's two types of applicants. There are those who are deeply immersed in the subject matter of bioinformatics or computational chemistry or computational biophysics, whether they be third-year undergrad students or graduate students or postdocs. Those are the ones that had a directionality to their educational experience, and they have clarity generally as to where they want to fit. And then there are those who are anthropologists who want to bring in a human element, to Shamir's point. Highly valuable, but maybe they went down a different career path educationally, but now they want to make the leap. I have an undergraduate degree in psychology and organizational behavior. I spent time in finance and strategy, and somehow I joined and co-founded an AI and drug discovery company, and now I'm the CEO of this company. So there is merit in the non-classical approach. We have hired musicians as data scientists who then self-code, learn to self-code. We brought on musicians to help on our design of our user interface for our internal. And so it really becomes, how do you tell your story depending on your directionality of your career, related or unrelated, if you are genuinely interested in joining that company. Your brand is the sum of all experiences that a person will have with you. So you've got to make sure that as quickly as possible, but as consistently as possible, you're able to tell that story as to why that company should choose you.
Tarun, would you like to add anything to that?
Sure. I think my panelists had a really strong points. I'll just keep it very short. But I think Shamir put up a fantastic point when he said you need to differentiate your application, or you need to differentiate yourself. And there are various ways I have seen applicants differentiate. I mean, some of them really use social media really well. And by social media, I mean LinkedIn, like, you know, putting yourself out there and sharing the work you've done. It can be small research, but positioning it how it can have an impact in the long term, or how it can add value to the company that you are applying for. Another thing that really works well is understanding the company that you're applying for. Not all companies are the same. You may want to keep different resumes. Some are focusing on, say, oncology. You want to focus what you've done in that area. Some are focusing on, say, cardiovascular. You cannot have the same kind of research for both. It's again points to differentiate yourself, because if I'm reading something where I'm working for an oncology company and someone has that experience, yes, he'll be weighed slightly higher. And of course, it doesn't mean that you have to know everything. What it means is how you can position yourself for that role, that yes, you are the person for this job. And then the other point that both Nahid and Shamir put forward is on the teamwork side of it. I think I would love to have a team player on my team. So someone who can work well with others, and there are ways you can distinguish, or there are ways you can focus on in the interviews and in the overall process. And someone who can show that impact, even if he's not done a lot of research, but is willing to learn and willing to make an impact. So I think these are certain ways where you can really differentiate yourself. And that's the key point I would take away from the conversation.
Thank you. I think it was amazing that we all discussed all of this, because if I have to summarize, there was so much value in all of the conversations that we have had. It's really difficult to summarize all of it, but I do have a few points that I would want to highlight here. So all of you being leaders and being able to do a lot of training in this manner, where we are talking about integrating other players into our team, and then it requires to have that sort of understanding where this process is get simpler. Like, it should not be more complex, then it becomes challenging. So what I've heard from Shamir is that there are some skills and that is required, but there are some other soft skills also which are required. So at times, I think it helps to think them through and have a strategy, or think about the things that could happen. And that kind of vision comes from this sort of understanding, where you're telling that these are important things that we have to consider. So that I think that gives us a lot of value to the students who are listening here and maybe thinking that, okay, maybe this is a X scenario, and if I have to put myself in that X scenario, what are the different outcomes, different people, different thinking that I could address while being in a group, while being a team player. So I think it requires a lot of thinking in that context, and being able to understand about a company. I saw that like one thing that I would wanted to mention, especially for a big pharma, or like when we would want to understand that what is actually going on there. So the difference, like what we see in research labs, many times we kind of look for the PI, and then we find the articles, and then we find that the methods that they've been using. So it's really kind of easy for a student to investigate further about a research lab, and look for the PI and go for like an internship or something like that. But when it comes to a company, the openness there is limited. So I mean, for us to understand that how bioinformatics differs in a company versus a research is a gap, or is also a challenge. So Dr. Shamir, what do you think about this?
Yeah, sure. So, there is nothing like a perfect candidate, right? Like we always have, there is a human element to hiring for that matter. And we don't expect, everybody to know everything. And within our data science and AI group, we have a significant section of our group is actually dedicated to learning and development. So you will bring certain skills to the table, but additional skills, whether let's say you have some background in statistical modeling, but you want to learn computer vision, we will have courses, or we will immerse you within a team that actually doing computer vision day in, day out. So we need to make sure that you will come in, and then you can build your your technology portfolio, your skills like that. So that's always possible in a pharma company like in a company like ours, or other players in the industry as well. And then the other part is like what we have to understand here is, if you take bioinformatics as a whole, like what some of these parts of bioinformatics we will do internally. Some of these would be done by CRO, some of this would be done by other types of companies, smaller companies that are actually hyper focused on that particular type of analytics. And then what you will do internally might be still at the bleeding edge. So you have to get ready for that. It's going to be a little bit different than a traditional academic setting where you will keep on analyzing or improving your pipeline. Because we need to move the needle. We need to push the boundaries, because anything that you do right now is actually like, if you are in a more R&D focused role, it will be you're potentially contributing towards a drug that would eventually get developed 5 to 10 years down the line. So you're working backwards, right? Like you're not going to get an FDA approval, in this year or this month for an R&D stage molecule right now. So you will basically contribute that. You're working towards what could happen in 5 to 10 years. And if each and every one of us think about where bioinformatics was 5 to 10 years ago versus now, we can literally see it growing, right? Like, whether it's access to high performance computing or more algorithms, more methods, we see that. So we just be future ready, just be ready to sink in. And if, that's what I would say, and go with an open mind. That was Tarun's point, right? Like, if you're good at certain skill, whether it's writing, communications, social media, or even as simple as making videos, have a catalog of your work somewhere so that we can see that we can understand your skills. Okay, you bring insert something that's unique, and then you want to come in and become a data scientist or running pipelines like this. This can be done, that's for sure. And one more last point I could make here is that the industry is seeing an influx of career in this space, right? Like, and Nahid also mentioned about this. We have, initially it was just data scientist. Now it's data engineer, there's two separate roles. Now we have, we used to have, DevOps to support the whole development team. Now we have MLOps, AIOps. Now, down the lane, 5 years on, I don't know, maybe there is a bioinformatics ops, I don't know. I'm like, so there are new things that would eventually evolve, and you can shape up your career towards that. So be open. And one advice that I could give to the students is that just because you have strong biology, you doesn't code, don't think you cannot work in bioinformatics, because we need a lot of analysts. We need a lot of people to do biocuration. We need a lot of people to validate the algorithm. We need a lot of test engineers. So there is nothing like, okay, a good bioinformatics job means, database programming and advanced machine learning. No, it's fundamentally, it's communication, writing skills, your like I said, stakeholder engagement, these are the skills. And on top of that, be good at one thing, either it's programming, either it's generics, either it's curating variants. It doesn't matter. There is always an opportunity. And I personally don't believe, even with the influx of all of these advanced machine learning or applied machine learning, I'm someone who's doing this I started doing it in 2002, maybe 2001. It's going to be 20 years. I've seen that applied AI in biomedicine for the last roughly 20 years. Don't worry, algorithms are not going to take your job. Don't worry about that. Don't worry about it. It's good to have that good algorithm development skill, but we still need curators. We still need developers and engineers and all. So yeah, so that would be my take on it.
Those are some fantastic points, Shamir. I would love to add a bit here. And, one of the things I also see when people transition from research to the corporate side is I think the people succeed when they are able to relate to what makes the company succeed. For instance, when you are doing a research, your focus is on, okay, let me focus on this particular aspect of the value chain, and you forget the downstream value chain. And the downstream is your clinical development, your commercialization, and bringing the product to market where you can help patients, right? So the more you converse with your counterparts downstream, like talking to clinicians, how do you decide the endpoints? What patient segments are we targeting? How is this research adding value downstream? And then also talking to the commercialization folks, like what is the patient need? And then, just having that dialogue with your colleagues across the value chain, that really helps when you're entering a company from the research side. So yeah.
And if I may, just to pick up and amplify on what Tarun has just said there, my experience is often that with people transitioning from science to industry, that they are very focused on the ground truth and the proximate deviation from a ground truth, and sometimes, and most often in an entrepreneurial environment, certainly building a company, there's a lot of ambiguity and a huge amount of abstraction is required to be successful. And perhaps this is most simply characterized with fixed versus growth mindset. I think what Tarun's saying, the people that are successful in reaching out to understand downstream the shape of what's coming, those are people that are required to have this growth mindset. I think, to really benefit in the future, that is a key attribute, I'd say.
I don't have too much more to add, and I want to make sure that if there are any other questions, we can go to that. But the only thing that I would close out with is know what you don't know as well, and go seek out that knowledge. Education is the best competitive differentiator, and it doesn't have to come through classical means. Coursera, edX, there are so many different, MIT with the Sloan management they have and the relationship with Coursera. I have no affiliation with MIT. I've just taken a bunch of courses and I have 4 more lined up for the rest of the year. Within the company or outside of the company, just continue to see trends that you like, follow some, and then create your own. And the only way that I think that's possible is through A, continuous education, and B, application and testing those things out in the context of what works, what doesn't work, and continuing to iterate. You can't call yourself a ML scientist, a data scientist, if you're not willing or able to Bayesian update your own mental model based on things that are happening in the industry. And so as students, just continue to, whether you're a high school student or you're 65 years old and looking to get into a new industry, or myself, which continuing to try and learn and stay ahead of stuff, just continue to educate.
So thank you, everyone. This really kind of like what I heard, some of the things, and I heard very different things, So Anthony mentioned they have an established apprenticeship program with a particular university, and I'm imagining you're kind of trying to build that out further. And then, Shamir mentioned that he has interviewed a lot of applicants from around the world. There are specific positions and specific skills that they're looking for, but there's a lot of this corporate structure on top of basic skills. And then I heard also from Nahid, as a smaller company, how important it is to kind of realize the vision and kind of align yourself with that vision and kind of highlight how you can contribute to that and grow and continue and expand. And then Tarun also mentioned how, understanding the domain and kind of like showcasing what you have done in that domain is very important. So that brings me to this question of relationship with universities. You can obviously build your own ecosystem, right? You can participate in research and showcase what your company is doing in that space. But in many cases, students don't come from nowhere, right? They're already a part of an ecosystem. They're already a part of something that they've been led to believe or to kind of envision themselves, right? And those examples that they have in front of them and the pathway that they've went through that kind of shows them what they might be possible, might be able to achieve. So my question is really, how can universities prepare students for careers that you see in industry? Specifically for companies that hire bioinformaticians or from those backgrounds. How can faculty at universities, how can the administration at the universities potentially work with companies like yours to establish that pathway and kind of provide that future for their students?
I can take that one first if you'd like. I don't come from a research institution, so for profit, but we have at the moment relationships with over 220 research institutions globally as part of our Cyclica Academic Partnership Program. And we open up, and it's acronym CAP, to work with academic and research institutions for two reasons. Number one, we make available our computational drug discovery platform available to PIs, principal investigators within those research institutions at no cost to them in the pursuit of advancement of basic science and the translation into potentially commercial viable assets that could maybe one day become drugs. And the second and most important reason is to build brand recognition with the next wave of students and researchers who will be trained under those PIs and may become available into the marketplace eventually, whether they be at big pharma companies like AZ, genomics companies like with Anthony, or they continue to remain within academia and postdocs and then become a PI themselves. How do we help train or affect behavioral change in the minds of many scientists on what is possible with computational approaches, both bioinformatics, physics-based, AI, machine learning, deep learning. So we think a lot about that, and we've instituted that CAP program, and it's been highly, highly successful. Then there's another type of relationship, and I'll point to a few examples. I think Boston does this exceptionally well, and so I'm sure Tarun will talk about how, and I'm not picking on Boston again, I have no affinity, I'm Canadian, but like Boston has done this exceptionally well. Stanford has done this exceptionally well. I think Stanford, I saw a release recently in the past 24 hours where it stated that over the past decade, they've started 680 companies, and the total market cap of those companies is close to 20 billion or $50 billion or something like that, just from startup companies that have come out of Stanford. And so what does that then mean? That generally means is that those companies are created with brains that have been educated in those research institutions that then go raise venture funding or funding from other environment strategic partners, go translate that research, hire people, create actual economy and jobs, generally from the ecosystem as the term was used, and probably likely from their academic institution that they came from. And so there becomes this virtual cycle of training, educating, and then deploying into the marketplace. And there are a couple areas in the world that have done this really, really well. I think Canada, and specifically the beltway between the University of Waterloo and like McGill University in Montreal, have done this very well, especially at the intersection of experimental sciences like biomedical sciences, as well as computational and computer science, AI, etc. With University of Waterloo being a very, very strong school at that frontier. And right at the epicenter is the Vector Institute in Toronto, and then MILA, the Montreal Machine Learning Institute in Montreal. And what they've done is brought in the hospital networks, the research institutions with industry and government funding to not only train the next wave of students, but to provide them with the basic pillars to enable the commercialization or translation of that research into commercially viable entities. And so you're looking now at where is the next frontier of innovation? Yes, absolutely Boston with MIT and Harvard and Northeastern, which is really coming up. And then you have in California, but you have a lot of small little sectors that are growing globally. And I'd point to what's happening in Toronto and in Canada in our space as probably the vectors of influence pointing there.
Yes, thanks, Elia. Just echoing the model that Nahid described, what we're trying to do in networking microbiome science is connecting the leading research establishments that are driving the art and the science with the enterprises that are seeking to exploit that. We're focused not so much on drug discovery in the short term, although that's increasingly part of our focus, but helping industry participants in nutrition and food and nutrition, cosmetics and personal care, and ag bio to network understanding of microbiome science into their models, and on our platform connecting the research establishments with the enterprises so they can collaborate with us, if you like, as the glue in the middle around some particular projects. We're also often sponsoring PhD students within the research establishments. We describe them as knowledge partners that we're working with. And we already have quite a highly distributed business. We are in Cambridge in the UK, in the centre of London, King's Cross, what's called the Knowledge Quarter, next to the Alan Turing Institute, the Francis Crick Institute, Google DeepMind. We're in Manhattan in the New York Genome Center, in Potsdam on the Potsdam Science Park, in Paris in Microsoft's AI Factory, and also in Hyderabad, where we have 30 people already. So we're trying to create local partnerships with research establishments because we want to foster relationships and encourage candidates from those research establishments to come through to join us. But it's really important to us that we are creating clusters where humans can interact with humans, not if in this quasi-virtual world in which we still inhabit. It's not necessary to be together with a mentor or whatever every single day, but it's absolutely germane to our evolution and building of our business that we afford people the opportunities to interact with other humans on a frequent and regular basis. So that's driving our model.
And maybe I could ask actually Tarun to answer next, and together with that, if you could kind of address this question, how do organizations deal with converging business and scientific rigor? So business interests and scientific rigor together without damaging either.
Wow, that's a tough one. So there are a lot of conversations around that. How can we have best of the both worlds and make sure that the research we are doing, that's one hand, and the commercialization that we're doing, that's the other hand. And how can we succeed by joining both hands, right? And often the trick here is communication. I mean, an open communication where you're empathizing with the other party, that yes, I understand what you're doing. Not blaming the other party if there is failure. Failures are bound to come. The failure can be from the research side, it can be from the business side. And then I think that's where the role of the CEO, and I think Anthony and Nahid can vouch in there as well. How can CEO play that role where he showcases that yes, the business side is important as well as the research side, and collectively is when we win. So making sure that the whatever activity we are doing, by activity I mean it can be experimental research, it can be understanding what is happening from a clinical perspective and also understanding what is happening from the business perspective, and mentioning how all of these tie up together. And I've seen that, sometimes the business perspective is that, hey, I know what I'm doing, I don't need to understand. You are a researcher, you don't understand this business. So just aligning both parties there. And it's a good question, because often it can be detrimental for the organization. So what worked for me when I was working in this area was having sit-togethers with both parties together and somehow being that bridge that can help communicate, because you need to understand both sides of the business to be a good mediator here. And I think I would love to hear even Nahid and Anthony's perspective here, because as CEOs, they are doing it day in and day out.
Tyrone, if I may just go directly to that, you've just pinpointed the primary job of the CEO, both internally and externally, is to be translator and modulator of information delivered from different sources, speaking perhaps all English, but fundamentally different languages towards a common goal. And I think at this early stage in the evolution of our industry, where we are bringing together individuals from backgrounds, educational backgrounds, and experiential backgrounds to a particular focused endeavor from a range of different environments, translation is the core skill. And that's the other reason, by the way, why we're so focused on this apprenticeship approach, because we're trying to create the mechanisms to enable people to understand the voices from the range of sources earlier in their careers, because they get too siloed, particularly perhaps most often with PhD postdocs, very focused and without the sort of constant perturbation, if you like, that it forces the individual to explore the broader environment beyond the core focus. That becomes increasingly difficult later in a career.
Yeah, not much. Again, you're getting frontline seats to some very thoughtful perspective. There's not much more I can add to that, but I don't also want to remain mute on this important topic. I would say, unlike any other time in history, today there's a convergence of skill sets based on a bunch of stuff that now exists that did not exist even 10 years ago. Again, I go back to the availability of high quality, a lot of high quantum and relevant data that is, when trained with new algorithms like AI or ML, deep learning, what have you, and powered by new infrastructure, cloud computing, or even that which is coming down the pipeline, you now have this convergence of opportunity. And so unlike before, where chemists used to talk to chemists, biologists used to talk to biologists, clinical scientists used to talk to clinical scientists, you're now seeing pharma generally over the past number of years create an environment where these people are sitting together, sharing ideas, and therefore incubating converged approaches. So med chemists aren't only talking to med chemists. I think Novartis has come out and actually led the charge in terms of at least how they branded themselves, whether practically, I have no idea, but they brought chemists and biologists and computational folks sitting together. And so as I think about that model and vis-a-vis Cyclica, we have internship programs where we bring on biomedical scientists, bioengineers, computational chemists, but then expose them to different areas within the organization. So we have a group of applied scientists, which is what we call them. There are drug discovery hunters, med chemists, organic chemists, biochemists, biologists, folks that have lived at the bench to do a bunch of stuff, but who have a propensity towards computational techniques or data science techniques. Now, whether or not they trained that way as part of their education, formal education, or they did it on the side, what we've brought in is a group of people who are really curious about not doing it the classical way. And so I would continue to encourage, the company that you want to join, are they able to offer you that, whether it be through formal training or apprenticeship programs like Anthony's company? We do very similar tag teams, bring the teams together and make sure it is interwoven and not separate. But then I would continue to encourage the group of students here, no matter where you are in your career, to continue to seek out that level of information bolted onto the thing that you spike in the most, what you do and what you're most passionate about, but continue to add on to it. And from a company standpoint, make sure that they're able to fulfill that curiosity, that they're able to scratch that curiosity itch for you. As a result, you're able to contribute back to that company, bring a new perspective, but then also leave much more, I'd say, broadly, and like you'll be able to fight the curse of kind of peripheral vision or obfuscating peripheral vision.
Thank you, Nahid. I think all of these thoughts are, very helpful. So, that brings us to the next question, which is on behalf of the community. So we get to interact with a lot of faculty from different universities, and they would want to maybe get involved in a research which is more directed towards the industry. So the question that we get is that how can we get at level to an industrial grade research? And we do understand that there's a gap, but when we think about a community, so I think there is a possibility to leverage that community. So maybe Shamir, I can ask you in that aspect of how do you think AstraZeneca as a company has looked into to address this, to utilize maybe a community or make them bridge this gap?
Sure. Yeah, and then I can also add a quick point for the previous question that the co-panelists were discussing. So we're always looking for talent. We always have internship, co-op, other types of positions. You can always look into our data science and AI career page and apply directly. And then also you can reach out to potential team members, including myself, through LinkedIn or Twitter or whatever is your preferred social media. And we're always looking for collaborations as well. And coming back to the collaboration specifically, so we do have both structured and sort of ad hoc collaborative engagement throughout with our community, in the research community here. And that means, like, for example, we do collaborate with Cambridge mathematics program. So we do research collaboration. We have sort of a co-founder of the Cambridge Center for AI in Medicine in the UK. We have collaborations with Mila, with Broad Institute, several other leading research groups that we collaborate with, and those are like sort of structured. So that's one approach that we're taking. And then also we have we strongly know that we won't be able to solve all of the problems that we have, right? Like, so we do have what you call as a platform called CoSolve. Like, we need communities input to solve some of our grand challenging problems, actually. And if you can look for AstraZeneca CoSolve platform, it's led by a friend who leads the emerging innovation unit, and you can see a number of projects or problems that we are actually openly looking for collaborators. So that's a second approach. And the third one is, like for example, my group primarily doing machine learning research, and we are always looking for collaborators. We know the problem is these are some of the grand challenges in the industry. We need to work together. So we're always looking for collaborators. So that's on an individual project level or an individual research engagement level. We can definitely think about. So it's the structured programs, there is CoSolve, and then also between potentially interested research groups can collaborate as well. Yeah.
That's great to know. Anthony, do you have something for the, researchers like faculty or early researchers to collaborate on? Because many times what we have heard from them is that they wanted to work on exciting projects, they have students, but the research idea is something that does not align well or is not maybe properly structured at times. So maybe at an industry level, if that sort of help or at least guidance is there, that could actually lead to some great outcomes with whatever resources that they have.
Well, thanks, Mohit. I think it's challenging to give you a coherent answer in describing sort of point solutions. I think we're trying to look at this at the macroscopic level by trying to understand the incentives of particular research establishments, what drives their interest and focus in engaging with a particular enterprise, or what their research niche focus is, and whether we can match that with a particular enterprise that's looking at, you know, the gap in its own capability to be filled with that understanding. And we're actually trying to instantiate on our platform mechanisms for matching to enable that, so that we understand the incentives of the research establishments, we understand the incentives of the enterprises, and we can seek to match. That's a challenging endeavor, but it's already bearing fruit, because we're selling our software application, the eData Scientists, into large enterprises, and they're asking us to network data from research establishments on the other side. So the beginning of this ecosystem orchestration opportunity, you know, we're seeing that now. But I think we're still some significant way from being able to, you know, say to a particular researcher within a particular knowledge partner or research establishment, you know, look, you're doing interesting work. This enterprise is interested in perhaps collaborating with you. I do see over the course of the next 5 to 10 years, the evolution of more clever data-orientated marketplace structures where we do match research establishments with enterprises, because that's necessary for the acceleration of the science that we're all involved in at the moment.
Thank you. So one question that I have, especially maybe if Nahi, we can answer that. So we heard about top institutions being one of those places where the companies look forward for innovation as well as for students. So there are also, we got to work with a lot of students from Africa, especially we have a collaboration in Nigeria. And there are other countries where students are there who are equally capable, but they are not exposed to these opportunities. But the things have like kind of changed with the COVID, the silver lining that many things are now online and they get an opportunity. So do you have anything for students like that who are coming from maybe like so-called third world countries where the education system is not as structured or as good as other countries comparatively?
Good science happens all over the world. You don't have to have a MIT or an Oxford or a Cambridge or University of Toronto or a Berkeley logo next to your resume. Now, so that is a firm belief. Secondly, from our perspective, you will quickly suss out from the company that you're applying to whether or not they have that ethical alignment. Look at the people on their website. Are they generally building a company that is of like-minded people to them or have similar degrees from similar types of institutions? Or is it a little bit more diverse? And are they open to that belief? So I think, unfortunately, I think a large percentage of companies generally, especially in North America and many in Europe, will go and hire from institutions that they were trained at or that they know very closely. We've tried to push on that belief. There are complexities in bringing on, as a Canadian or a US or European company, bringing on co-ops or employees in other countries. So there is a hurdle that companies need to overcome to justify bringing on a student, whether it be in Nigeria or in Rwanda or in the Philippines or in Papua New Guinea. And that's tax and employment law and all these things. And so how do you convince the company that you are the best person for this and for them to make that investment? So that's, from the role of an individual applying to the company. Now there's the role of the company and what they need to do. And I continue to encourage companies, especially early stage, that if it's not us that is going, especially the leaders in the industry, if we're not willing to think differently, then it's never going to happen. So I spend a lot of my time globally outside of North America and Western societies. And I've spent a lot of time in Asia, in many of the emerging markets. I've spent a lot of time in Africa. My family is East African, Kenya and Tanzania. And I was at the Next Einstein Forum in Kigali, Rwanda in 2018. And I got to speak to, on behalf of the Chancellor of the University of Rwanda, a professor at Carnegie Mellon. and the managing director of Elsevier, we got on stage to speak with 2,000 STEM students at the University of Rwanda to encourage them to continue to pursue their passions, to help them think about how to brand themselves to companies that they may want to join that aren't doing business in their country. But then also my rallying cry was, how do we as companies that have the resources available, how do we continue to invest in those students, whether it be through technology access, education, exposure, or through internships or other environments to bring them over? So I'd say we're not nearly perfect, but it's worth the fight. We have, as our director of drug design platform team, he was educated in South Africa, right? Like, we have students that we've brought. We have students from India that we have brought over to Canada, students from China that we brought in over from China to study and to work at Cyclica. Now, that mindset has to shift from a lot of companies to enable more open environments. But from a student perspective, that's not your business. Your business is continue to fight and differentiate. And if you want it, just go tell a really good story as to why that company should choose you over somebody that's in their own backyard.
Thank you. I think that is a lot of courage to all of the students here. Now, maybe Ilya, do you have something to add?
Yeah,
Tarun
so
has to say something. There was a hand raised by Tarun. Tarun, did you wanted to add something to this? And especially for the students in India?
No, I was just clapping. I mean, I didn't want to raise hand.
Okay.
I agree. I agreed with the point that Nahid said. I won't take much time, but yes, I think I'm in complete agreement with Nahid here on both the points. The world is your oyster. I think it does not matter these days if you are bringing value. It does not matter where you're coming from, and the focus has to be on showcasing how can you, make that impact, create that value for the organization that you want to join in. So focus on that rather than, again, there are controllables in life and then there are uncontrollables. So wherever you've already studied, it's an uncontrollable now. So let's not focus there, right? So I only want to focus on what I can control. What I can control is the next steps, and that is maximizing the value that I can bring on the table. Just focus on that. Don't even think where you're coming from. And then second is, again, in agreement with Nahid, it also matters what the philosophy of the company is. If the company is someone they only want to recruit from a certain places, don't apply there. I mean, that's their loss, not your loss. So figure it out and go for that opportunity that you think you're capable of. Don't let the past or don't let where you're from let you discourage. That's one thing I definitely want to put on the table.
So I just want to jump in here, and I want to mention that it's been really a pleasure to hear all of your perspectives on these important topics from all of our participants. We want to ask you for just your final remarks, and we want to then move on to the poster competition. But I do want to ask everyone if you want to add, a minute or two of final remarks before we move on. Thank you.
I'll kick it off, as I just want to add one more comment to this important topic that Tarun and I just covered. I'll give a quick example and then I'll conclude. Now more than ever, especially in a ongoing post, whatever you want to call it, COVID environment, what's become clear to companies is that talent is global. And you're seeing not only talent, but venture capital institutions are funding. They used to fund in their own backyard because why not? And now they're saying, well, let's go fund globally. And where capital goes, then teams grow and things are leveling off to an extent where companies are looking to hire. And so now more than ever, ramp up your effort to go find those institutions that admire, or that you are most aligned with, that you admire, that you are scientifically passionate about. I'll give a great example of a student who approached Cyclica. She's now on our machine learning team. She's been with us for 2.5 years, but she came from Cuba. We had no idea who this person was. We had no idea that Cuba had a strong systems biology and bioinformatics division, because we just weren't spending time scouring the Cuban market for talent. But this individual found a way to somebody important within the Cyclica environment, built a relationship with this individual, and that individual pinged me and said, I think you need to take this person seriously, have a conversation. And within a few weeks, we went through what in Canada is called the labor market investment analysis. We invested a lot of time and capital out of our pocket to bring this person over to Cuba and manage their entire visa application, whereby we could have saved a lot of time and a lot of effort to just go in our own backyard because that individual was the right person for us. But how did they go about it? What they did not do was ping me on LinkedIn and say, hey, I'm interested in joining Cyclica. Can I have an interview? Because I'm the last person that is going to know what to do for a specific contributor on a team. So my encouragement is LinkedIn is an important tool. And since sitting here, 20 people have asked me to invite over LinkedIn. Awesome. I've accepted all of you. But if you're interested in joining Cyclica, understand what we're doing, understand who within the company is the most important person to build a relationship with, build a relationship with them. It will take time. And that's the way you differentiate yourself beyond just applying to a job online. And so it takes effort, it takes smarts, it takes strategy, and it takes the will to allow the time to come at you. So if I'm going to conclude, my feedback to all of you is use the resources that are available to you as a overall strategy, build that strategy, and be smart about how you go about it, because it is possible. And I can give you dozens of examples of how we've gone about it. But each one of those, that individual came to us in a very unique way.
Thank you very much. Anthony, do you have any closing remarks?
I think, understand your why as an individual. Why are you doing what you're doing? What is your motivation? What is your end goal? End goal in terms of what fulfills you, rather than a narrow short-term research objective, and then try and match your why with the why of the organization that you want to work with. Because if you do that, then you will liberate your talents and you'll deliver something to the enterprise you're working with. I think just echoing what Nahid says, the COVID pandemic has caused us at Eagle Genomics to build a highly distributed global business because there was just no other way. And actually, we've discovered that that's a competitive advantage. We're not going to go back to the old. We're not going to consolidate back to the small, narrow geographic focus we had previously. So there never has been a better time. But do your homework. I think the panelists have said that before. Understand your why and match it with the why of the enterprise you're seeking to work with. And then I think alchemy can be liberated with that.
Okay, thank you. Shamir, would you like to add something?
Sure, thank you. So I strongly believe, irrespective of all the advances that we have right now, whether it's in, within data, machine intelligence, algorithms, robotics, we are still, not close to that sort of a dream of getting that right drug to right patient that would lead to an optimal outcome and and least side effect. We are not yet there. We are on a path towards that, and we are all in this journey together. There are plenty of career opportunity, and the field is just getting more exciting because we are now moved from mono-omic data set to multi-omics, and there is more to come. If you look at the biological aspects behind this. So it's a really exciting field, and we need all hands on the deck. That's what I want to kind of put together as my last comment.
Thank you. And Tarun, maybe you could summarize.
I think most of my panelists have already covered the major points. I'll just keep it on the philosophical side, where I would say keep that inquisitiveness quotient on. I mean, often what makes the difference in the longer run is asking that one question from someone who can help you. And more often that, I have realized in my career is that people are willing to help. People are willing to answer those questions, even if they look stupid to you. So, keep on learning. There is no limit to that. Beef up on that knowledge. And reach out to people where you face hurdles, and you never know who can be your guardian angel and can help you. So that is something that even Nahid said, someone reached out to him, he took that call, and often, you'll find walls as well, but you'll also find your guardian angels. So just keep on doing that. I'm happy to, answer more questions on LinkedIn as well. I'll post my LinkedIn ID. I connect with people wherever needed. So happy to help where I can.
All right, great. Thank you very much, everyone. And thank you, Mohit, for moderating the panel. Would you like to kind of summarize what we've heard so we can transition away?
Yeah, thank you so much. I mean, we have had a wonderful session with all of you here. Thank you for your time. And I think we got to learn a lot from a student perspective, from a researcher's perspective, from in general, that what are the steps that are required for us to take next. And we also learn about the internship opportunities that each of the companies have to offer, along with other opportunities and skill sets that you are looking forward to. So I think it was great for all of us. And I would like next move to our much awaited poster presentation session, where students have submitted a lot of, they did a lot of work, and they shared their abstracts and their posters, and they presented with a video presentation. So I heard also that presentation skill sets are kind of very important, and how do you articulate yourself to express. So the expression part, and to do that, we encourage people to do video presentations, and we got a lot of those, which was judged by a panel. So now we will just move to that. And I would again like to thank all of you for being part of this. And now back to the presentations. Thank you.
All right. Yes. Thank you very much, everyone, for joining this panel, for sharing, to all the panelists and the speakers. We do have a poster presentation. So what we asked everyone to do, we had over 100 students that have worked on research projects over the past several months. And what we asked them to do is to kind of find a question to ask. Thank you, everyone from the panel. Really appreciate you joining, and hope to continue this conversation beyond the symposium, obviously. And so what we want to do now is kind of offer a perspective from our industry, from our research judges. We had Professor Sona Vasudevan from Georgetown University Medical Center, Dr. Chris Taylor, the director of the Bioinformatics Core at LBRN, Dr. Philip Campbell from Carnegie Mellon, Julia Panoff from Haifa University, the associate director of the Tauber Bioinformatics Research Center, Dr. Giuliano Chavez from University of Chicago, Dr. Abhishek Sengupta from Amity University, and Dr. Harpreet Kaur, who is our research consultant and Omics Logic curriculum developer, to evaluate all of the submissions. And I would like to ask Sonalika to please go through the scoring and explain again how was the scoring done, and help us understand who the top posters were. Sonalika.
Thank you so much, Ilya. Let me just quickly share my screen and tell what our judging criteria was. Okay, so this was the form that was given out to our judges, and the judges were as explained by Ilya. So here we had a few points on the basis of which the judges have given the score to each and every participant. The first was based on the introduction score, and then the methods and results, how perfectly were they explained, and then the discussion and the conclusion score based on the discussions and the presentation quality. And we had a poster presentation submission page of each and every participant, where we had all the poster submissions laid out. Let me just quickly pull out the page and show it to all of you as well. And based on that, we have our final winners. So we received a number of abstracts, and out of all of them, we submitted 20, we accepted 20% of them, and we enlisted them on our poster presentation submission page, which was with our judges since day before yesterday, and they were evaluating it. And we had the video recordings and also the poster PDFs of each and every poster. Let me just share the link with you all as well to have a glimpse of it. Thank you so much. And based on that, we have our final winners. Our first winner is Kira Tang with a score of 84 points. Our second winner is Dhruv Mehra with a score of 82 points. And our third winner is Andrew Lee with a score of 80 points. Though it was a very close call, the difference was way too less, but yes. So congratulations, Kira Tang, Dhruv Mehra, and Andrew Lee. And just to make sure, I'm not sure whether they are present here or not, but yes, so we have Dhruv with us. Ilya, first of all, over to you. Would you like to say a few words, and then we can have a word from our winners?
Yes, so I wanted to mention that we also have a special category called the Citizen Science, and that is related to everybody's scores, not just the judges. And so the top, I'll just read out the top three, but only the top one actually gets the prize. So Rutvi got 245 points, Aditayo 205, and then Supantha Day got 153. So a special mention for them as well. But Rutvi, if you could please add her, she's a special category for Citizen Science Prize. And yeah, would you like to pass it on to them to share a few comments?
Thank you so much, Ilya. Congratulations, Rutvi Vaja as well. Dhruv, I think you are here. So could you please take over the stage and say a few words? Over to you, Dhruv.
Hello. I hope I'm audible.
Yes, Dhruv, you're audible. And also, please tell us something about your project, what you worked on, in a brief way, maybe. Thank you.
Okay. So, I hope I'm visible and audible. First of all, I'll thank all the judges for this award, especially. And talking about the project, so I've been working on this project since March, I believe. So I started my journey at Pine Biotech in the March end itself, and then, after the vigorous training, I started the project. So talking about my interest, so it basically lies in the cancer research, and I'm more focused towards the translational part. So initially, I started, all the literature review, and then I found this the esophageal carcinoma, which is quite a dangerous type of carcinoma, and it's more prevalent in the India itself. And there was an altruistic motivation behind it. Like there were quite a few of my relatives who passed away due to this type of cancer. So that was a motivation for me to undertake this project. And then I started exploring, and I just saw it from a perspective of precision medicine, which I'm pretty much interested in. And then I moved forward and found the results, and I'm really happy to present it in this poster conference. I especially thank my mentors, Ilya sir, and Dr. Harpreet and Dr. Mohit for this opportunity. So thanks a lot.
Thank you, Dhruv. I wanted to ask, I know that Kira Tang is not present, but Gio is her mentor. Gio, would you like to kind of speak on her behalf?
Okay, so we have Kira Tang with us.
Okay, great. So maybe yes. Hi, Kira. Would you like to say a few words?
Sure. Hi, everyone. I'm Kira Tang, and I want to thank everyone for this wonderful opportunity and thank my mentor, Jepoliana Chavez, but also this couldn't have happened without my wonderful teammates. And this was based on the work that we interns did with Dr. Chavez over the summer. And it was really an eye-opening experience for all of us. For me, it was my first research project in bioinformatics, and I can't wait to see what the future holds.
Thank you so much, Kira, and congratulations once again. Now I would like to call on stage Andrew Lee. I can see he's online. I need to give the permissions for you to speak, right?
Yep.
Do you have
Hello.
Hi, Andrew.
So first off, thank you for the opportunity to have this award. I would like to thank all my mentors at Omnilogics that have helped me, and also at my internship at Columbia University. So my project was on autism and finding autism machines using machine learning. And that was my project that I worked on over the summer, which I had expanded with the bioinformatics from the fellowship program. Thank you.
Thank you so much, Andrew. Over to you, Ruthvi. Can you please tell us something about your project and your experience? And also, how do you feel? We can see you, Ruthvi.
Yeah, I wasn't able to unmute myself. So hello, everyone, and thank you so much for providing me this platform and this award. It means a lot to me. And for those who didn't know, this was my first bioinformatics project. So I initially started with Pine Biotech a year back, and I had zero knowledge of bioinformatics. So I've came a long way, and my mentors have pushed me a lot to do this. So this really means a lot to me. So thank you so much. And thank you for your constant guidance and support.
Thank you. And maybe we could just ask all of the mentors and also all of the winners to turn on their video. So we have Jepoliana Chavez, we have Mohit Mazumdar. Thank you. Kira Tang, Ruth Vivaja, Andrew Lee. Do we have Mohit? Okay. And so I don't know if it is possible for you somehow, Sonalika, to organize them so that they're in the same place.
Yes. So let me just first of all stop sharing my screen. All of you, please. Yes, let me just put them on spotlight as well. Let's have a screen capture and capture this beautiful moment.
Perfect.
Dr. Mo and Dr. Harpreet, are you available? Can you please, if you are here? Yes. Okay, it's coming in a very beautiful way. Thank you, Dr. Harpreet. Who's missing? Dr. Mohit is here. Ilya, we can see you. Okay, thank you. Perfect.
All right, thank you very much. So I know it's been a very long journey, hard work, and so you really deserve the prizes. So together with our sponsors, we have a symbolic prize, which will include, so we have, you will have continuous access to our platform. We hope that you do more with bioinformatics. You do have credits towards programs in the next 6 months, and $150 in cash awards from our collaborators. So we hope that you will use that money wisely, hopefully celebrate and buy yourself something nice. So again, thank you. We appreciate also all of the judges. So again, I want to say that, this has been an experience. I'll just share my screen again, where very established, senior people have reviewed your projects in detail. Many of them have seen, hundreds of projects. And so I think it's a real accomplishment that you were able to receive this recognition. Professor Sona Vasudevan, Dr. Chris Taylor, Dr. Philip Campbell, Ms. Julia Panoff, Dr. Gipoliano Chavez, Dr. Abhishek Sengupta, and Dr. Harpreet Kaur. You've heard their presentations over the past couple of days, hopefully, and you've seen kind of what they're all about. So really appreciate you taking the time to submit the projects. And we hope that you will, again, do much more with bioinformatics in the future. And as we heard today, there are many opportunities to get involved. All right, so thank you very much, everyone. And I will stop sharing. So in conclusion, right, Ruthvi, just let me check once again. Okay, Ruthvi, are you there? Or sorry, not Ruthvi, Sonalika. Sonalika?
Yes.
Yeah. Okay, so yeah, I'll pass it on back.
Yes, okay. So to all the participants, it was really great to have you all with us, and it was amazing to have such great speakers. And the last panel talk, it was just fantastic. Like, we got so much of knowledge, motivation, and inspiration. Like, words are less to tell what we felt. All of you might have felt that. So also I would like all the participants maybe to just switch on their cameras if they can. And let's take some screen captures. And we'll be back with another beautiful symposium next year. Thank you so much, Ojasvi. All the participants, can you please, okay, if you need time, a minute or so to comb your hair or style yourself, you can. Okay, I'm just wondering, how do I get all of them on one screen? Like, it's coming on top. Okay, spotlight for everyone, right, Ilya? Do I need to do that?
No, I don't think you need to do it. No, whoever's on video, you can just hide whoever's not on video.
Okay, perfect.
Yeah, don't spotlight anyone.
Okay, thank you so much, everyone. And also, maybe you can just share your experiences with us, and we'll share feedback forms. And yeah, the most important thing, the recordings of each and every speaker of each and every talk. And yes, the other much awaited and very important question, certification. I'm just getting questions on my personal messages for certification. So those who had registered themselves for the symposium, your certificates would be released by tomorrow, and they would be mailed to you. So you will get all your certificates on the mail, and also the recordings as well. And also all the details that were shared today about the institutions, each and everything. And don't forget to add our mail IDs, marketing@pine.bio, to your mail address, because you might skip it if it's not added to your address book. And also I'm sharing the mail IDs of Dr. Mohit Mazumdar and our mentor, Dr. Harpreet Kaur. It was great to have you all here with us. Now I would like to pass it on to Dr. Mohit Mazumdar, or maybe Ilya, to give a closing note.
All right, yes. So again, thank you very much. This was a great experience for all of us, and really appreciate all the work that our team has put in. All the people that have the same background like me have contributed. Some of them don't have the background, but that's okay. They also contributed. And so really appreciate the work that you put in. We know that it's been very long weekend. Nobody got any rest. But we are going to start the fall semester soon. So in any case, you weren't supposed to rest. So hopefully you all appreciated the speakers, the panelists. I think we heard a lot of very interesting comments and perspectives on what's happening in bioinformatics. And so with that, we are going to plan a session where all of the finalists and all of the top projects are going to be able to present their project in full. We heard today about the significance of the actual presentation skills and your ability to communicate your research results. So we're looking forward to that session. And during that session, we are going to also announce some of the upcoming programs that we have, starting with the Louisiana Biomedical Research Network, with Georgetown University, with other university partners that we have. So again, thank you very much for joining, and we hope to stay in touch with all of you. You can definitely connect with us, as Sonalika mentioned, by email, reaching out to us, and you all have the Symposium page that I think has all the information that you need to reach out to us. So again, thank you for joining, and hopefully enjoy the rest of this long weekend.